Everybody, hope everyone's refreshed, and welcome to everyone that's just joining. It gives me really great pleasure to introduce our last guest speaker for the day, which is Professor Sam Dupont, and he is joining us all the way from Sweden. So thank you, Sam, for making the time to join. So Sam currently works as an associate professor and senior lecturer at the University of Gothenburg, and he is also a consultant for the International Atomic Energy Agency. So he moves between Sweden and Monaco. And Sam's research interests lie with understanding the response of marine species to the multiple stresses associated with global change, using really exciting and novel experimental methods. And I've seen these in action, and they really are exciting. And Sam's science speaks for itself, but what really makes him special is his involvement and passion for several other programs, including capacity development programs that brings his exciting and high-level science to emerging researchers, students, and under-resourced countries. Sam is a passionate scientist and communicator, and he's also a really good teacher, and I'm really grateful for the opportunity to learn from him today, as I always am. So Sam, thank you so much for joining, and I'm going to hand over to you.
Thanks a lot, Carla. This is great to be here. I wish we could all be, in the same place and have the opportunity to talk more after this, but it's better than nothing to do that virtually, and I hope it's going to be fun for you. I was reflecting a little bit about what to present, and that was the title I sent back in the days, Ocean Acidification, A Question of Time, and then I realized maybe you're tired after the whole morning and after having your lunch, so I decided to do it a little bit less sciencey and a little bit more about what it takes to be a scientist today a little bit, and tell you a little bit about my own experience and how I finally developed my career and my lab. So I changed my title a little bit to How Did I End Up There? And I will tell you a little bit of my story. So originally I'm from Belgium, so this tiny country in the middle of Europe that you can actually see from space, like it's this little place here, because we have lights everywhere. And as a kid, I was not really connected to the ocean whatsoever. So I started being interested in a lot of things, but not really into biology or marine science. My goal really was to be Indiana Jones. I wanted to travel the world. I wanted to do an archaeologist and explore temples. But then time happened, and I realized that was not as exciting as I thought, watching movies and so on. But I decided to go to the uni anyway, but I had no idea what I wanted to do to start with. So my first attempt at university was really to try to be a civil engineer. So I did that for a couple of years. And my objective, I wanted to build rockets and space shuttles, basically. But something else happened at that time. So I was this young guy at the university for the first time, and I realized that there was a lot to do in student life, and I'm sure you're well aware of that. And I partied far too much, and I studied far too little, and I actually failed my second year of my civil engineering year, and I realized I'm not going to be able to make this. I was completely overwhelmed by what I was studying, and at that time I actually decided to quit university and do the other thing I was doing at the time, that was cooking. So I was actually cooking in a restaurant to make money to pay for uni, and I decided to do that. But then after a short talk with my parents, they convinced me that since I paid my tuition anyway, maybe I should try something else. And that's how I ended up studying biology. Because I had already credits in mathematics, physics, chemistry, and so on from engineering, I could combine my whole first year of biology in one semester. And what really drove me to biology was a really inspiring biology teacher and also an interest for social insects. So that was my goal. I wanted to study social insects. So I loved biology. It became really a passion. That's something that I really cared about. But again, nobody was working on social insects in my country. And I ended up being a botanist. So that's what I'm officially on my diploma. I'm a botanist, and I did my my graduate thesis on tomatoes and physiology of flowering. But at the same time, I was really curious. So I decided to take other courses out of my university, and I took these marine biology courses in this amazing place in France. So it's Roscoff Marine Station, one of the oldest marine stations in the world. And being there really opened my mind on what I wanted to do in the future. So I came to marine biology quite late. It was not really a childhood passion like many people have, but I really realized they are going during the tides. They have these amazing 10 meters tides in Hoskov. That was really something that I wanted to explore. And so I worked really hard to get a master's degree in marine biology, and after that got a grant to do my PhD. So in Belgium, you can propose your own subject. So I basically defended a project on evolution, basically. I was interested in that little guy. So that's a brittle star. Echinoderm became a big part of my life in the future, but I started working on those because this is the only species of echinoderm and brittle star that you find all around the world. So in any ecosystem, in any places, you're going to find those guys from the surface to the deep sea. So the question I tried to answer during my PhD was, how did that happen? Why make this species so special? And there are many different reasons for that, but that's basically what I did for my PhD. And really just to show you the importance of working hard, of course, but also the importance of luck. I went to my first big conference in 2000, and it was the International Echinoderm Conference in New Zealand. And I was there, and it was the day before my first big presentation. In many ways, I was a little bit nervous, but instead of working too hard on my presentation, I decided, okay, I'm going to relax a little bit today. And I went to the pub. And that little event, like going to the pub that day, is what shaped the rest of my life. I think that's always what I say to my students. Like, it's really important to focus on your work, on your science, and so on. But you also have to be a social animal if you want to be a successful scientist. So what I did that day really was to go to this pub, talking to other scientists, and I met a person that many years later will actually give me an amazing opportunity. So this guy is Mike Thorndike that you see on the picture here on the left. So we talked basically the whole evening there in that pub, drinking a lot of beers and talking about science, life, the universe, and everything. And then I didn't have any contact with him for a few years. I finished my PhD, and after my PhD, when I was looking for a postdoc, I applied for a position in Sweden at the Gothenburg Marine Research Center, where I'm still based today. And I didn't get it, but Mike, that you see on the picture here, was the director of the station at the time. And he remembered me from this day in New Zealand, where we drank far too many beers. And he basically called me and said, oh, I saw that you want to come here. If you're interested, I can give you a one-year position to, if you want to come, just come. And I jumped on the opportunity. I couldn't believe it. So I moved to Sweden with my wife at the time. And basically, I still remember my first day arriving in his office and say, okay, what do I do? What's the plan? And basically what Mike told me, which is overwhelming, was, okay, you do whatever you want and don't worry too much about money. So I had the best postdoc ever because I could really develop my own research question and take advantage of this amazing opportunity. And what I did was to work again on brittle stars because I had some expertise on that. But here in Sweden, we have a species that is truly amazing. The one you see on the picture here, it's called Amphira filiformis. It's living in the mud. And it has this amazing ability to regenerate. So most echinoderms do that. If you chop an arm, they will regrow it. But these guys, it's extremely fast. And so we developed together with Mike this project on stem cells and genomics to try to understand why these creatures are so good at regenerating where we are not. And basically, we share the same genes and so on. So that was a little bit of my question. So I worked on stem cells and so on. But deep inside, I really wanted to go back to ecology. And after 2-3 years of working on stem cells and regeneration, I decided to try to develop my own research project and try to go back to ecology. And I started by trying to culture these guys. So I was trying to develop strategies and methods to make larval culture of brittle stars, which is quite a challenging task. And again, I was extremely lucky because this guy came to visit. So his name is Lloyd Peck. And Lloyd Peck is a researcher working mostly in Antarctic and the Arctic. And he's from the British Antarctic Survey in Cambridge in the UK. And he was visiting the station, just stopping by, and he stopped in my lab and had a look at what I was doing, and I was culturing these brittle stars. I said, oh, that's really cool that you can do that. And have you ever heard about something called ocean acidification? And this was 2005, 2006, I don't really remember, but I never heard about this. At the time, there was barely anything new about that. That was a completely new subject. And he basically convinced me to give it a try and to try to test the impact of ocean acidification on brittle star larvae. I had to really learn a lot because at the time there was no best practices, there was very little literature, there was maybe 10 published papers on the subject at the time. But he told me, try that, it's going to be big. And he was right. And basically, he opened the door to my career, because started to work on something completely new is a chance that you won't have many times in your career. So what ocean acidification is, and I guess most of you know about it, but it's another consequence of carbon dioxide emission. So if you put more carbon dioxide into the atmosphere, a third is absorbed by the ocean, which is great. It's a great service that the ocean is providing us. And then it reacts with water and turns into carbonic acid. So that's basically, in a nutshell, what ocean acidification is. So we put more carbon dioxide in the atmosphere, it changed the carbonate chemistry of the ocean. And that's something that you can measure all around the world. So this is an example of monitoring. It's from the Mauna Loa station in Hawaii, where they've been monitoring carbon in the atmosphere for a while. And what you can see is that it's in red, it's increasing through time. So the concentration in the atmosphere is steadily increasing as a consequence of burning fossil fuel and so on. They also started to measure what was happening in the ocean, and that's the curve down there. So the concentration of CO2 is also increasing in the water, in the ocean. And then what's happening is that the pH is also decreasing in the water. So basically everywhere around the world, including in South Africa, the pH of the ocean is going down with change in the carbonate chemistry. So at the time that was already known. I know at the time when I started, we knew that it was happening and it will happen in the future, that the pH, the carbonate chemistry is changing. But what we didn't know was the biological impact. Seriously, at the time, there was like 10 papers where they were investigating that on corals, coccolithophores, and so on. But for the rest, nothing was known. But we had good reason to worry because the change associated with acidification are extremely fast. So it's happening within our lifetime, and it's extremely strong. And by strong, I mean it's deviating from the natural range of variability. So that's a different time scale where you can see that you have the pH, reconstructed pH in the ocean over the last 20 million years. And what you can see there is that the pH was fairly stable on average in the ocean, but is going down extremely quickly now. And what we expect is to have a 0.3, 0.4 pH decrease by the end of the century, and that would correspond to a doubling in acidity. So by the end of the century, the seawater will be 2 times more acidic. So a really good reason to worry about that. And also, so I did this first experiment, so very little was known. So I did something very simple. I collected brittle stars, spawned them, make larvae, and culture them in different pH. So for example, we compared 8.1 with, which is the average pH they experience today, and 7.1, which is not a big change, but is deviating from what they already experience today. And what we could see was really shocking. If you decrease the pH of the ocean just by 0.2 pH unit, they basically couldn't make it. Instead of these beautiful larvae that you have on the top here, you had a blob of cells trying to calcify, but couldn't make it, and they were all dead within a few days. So that was showing that acidification has the potential to basically lead to species and population extinction. I published my first paper in 2008, and again, being one of the first in the field, just by chance, by meeting the right persons at the right time, I became an expert, which is ridiculous, right? You can't really be an expert in anything in 2 years, but I started to get invited everywhere. And that gave me an opportunity to develop my research group, attract funds, and so on and so on. So I became part of this amazing ocean acidification community. And all together, we continued to collect data. And now we're reaching 10,000 publications, only like 15 years after I started. And overall, what you can see is that we have really good reason to worry, because if you try to summarize what is known, about 50% of all the marine animals that have been tested today are threatened by acidification. So showing kind of a negative response. So the science already today is extremely strong. If you check reports like the IPCC, like the latest that's been released only a few months ago, the phrasing that is used is really strong. It's like, we know as a fact, we are virtually certain, the highest level of scientific certainty, that ocean acidification will have dramatic consequences for marine species, marine ecosystem, and things that we care about. So all the ecosystem services that the ocean are providing to us. So at this stage, you really have to reflect a little bit, if you're a young or even an aging scientist like me, about what your science has to be about. And for me, I really evolved from this curious young researcher who just wanted to understand the ocean to this guy who really wanted to make a difference. And if you want to make a difference, you want your science to be focusing on the solutions. So we know acidification is a problem, but the next question is like, what can we do about it? And in a way, I always use this image. This is the situation in which we are today. So we are the little mouse facing like a huge challenge, which is the cat, which is climate change, environmental changes, loss of biodiversity. And when you're in a situation like that, there are three things you can do. You can wait and see, and that's, of course, a terrible idea. The second thing you can do is kill the cat. So if you can do that, that's the best option. You get rid of the problem. But if you can't kill the cat, the smart thing to do is to escape and hide and survive as long as you can. And then maybe gather forces, come back and kill the cat. And when it comes to climate change and acidification, these are exactly the options we have. We can do nothing, but then we're going to have to face dramatic consequences. We can kill the cat, which in that case means reduce or even stop carbon dioxide emissions. So it's called mitigation. You basically fix the problem. But we know it's going to take a lot of time. So what you can also do is escape, buy some time, and that's what is called adaptation. So these options can be make the marine ecosystem more resilient by protecting it, keeping the marine biodiversity, restore what has been destroyed, or change the way we do things. For example, adapt aquaculture to be more resilient to climate change. But none of these actions, none of these adaptation solutions are really fixing the problem. So we have to do both. We have to buy some time, but we also have to try to kill the cat, which is mitigation. And I will show you two examples of how we can actually make science that matters for these two different things. Because to be completely honest, we don't have time. I think it's been said so many times, and I guess the person that was the more blunt about it was the Swedish activist Greta Thunberg, when she says, the house is on fire. We should stop talking about these things. We should start acting on it. And that's exactly what we try to do now. So again, we have strong science saying we have a problem. We have solutions that we need to implement today, not in 10 years, today. And these two types of action and solutions are on one side, mitigation, kill the cat, or adaptation, buy some time. So when it comes to kill the cat, the mitigation, we have all the science we need. The science is really clear. We need to act. And really what needs to be done is change the way society is working. So reduce CO2 emissions, but also change the way we consume and so on. And a person that really inspired me for my research is Ken Caldeira. He's a researcher in Stanford in the US. And he said, people have to sacrifice a little bit of their short-term self-interest to help the world be a better place for the long term. And how you get people to do that is the most important research that can be done. So in other words, we can do research to understand better how to drive positive change in society. And one of the problems with the science we have is that we don't communicate it in an optimal way. And I don't know if you know Tim Minchin, this Australian comedian, but he said, the idea that the science of anthropogenic global warming or ocean acidification for that matter is controversial is a powerful indicator of the extent of our failure to communicate. So I reflected a lot on that personally. How, as a scientist, can I contribute better to the change? How can my science make a better impact? And I reflected, okay, as a scientist, why are we failing to communicate? And on one side is because we are not so good at it. We are poor communicator. That's not really our job. We are not trained for that. And on the other hand, we are nerds. So we get really excited about things that nobody cares about, including myself. I can talk to you forever about sea urchin larvae. Nobody cares, right? So if people don't really care about it, that means they won't listen, and that won't really drive the change you need. So the way we do science traditionally is that we have a problem, like ocean acidification. We do some science, we publish scientific paper, we create some information, and we pass that to society. We transfer that to policymakers, industries, and so on, and we hope for the best. It's called the science supply paradigm. Unfortunately, that doesn't work. I think it doesn't just work to just send scientific publication, even if it's like explained in a simpler way and hope for the best, because nobody cares. There is no emotional relationship with the kind of science we tend to do. So we imagined with some colleagues how we could do that differently. And we thought we have to move from the science we do today, which is located in what we call the zone of complexity. So we made like these two axes. One is the scientific uncertainty, and on the other one is how relevant it is for society. So most of the time, the science we do is complicated and on things that nobody cares about. And we argue that if we really want to drive change, we need to do science that is in that zone, like simpler science on things that people actually can relate to. So to test this idea, we decided to actually do an experiment on something that people really care about. And in Sweden, people really care about seafood. And what you see on the picture here, the shrimp sandwich is really a traditional dish. You find that everywhere in every restaurant in Sweden. So we thought if we can actually show that ocean acidification has an impact on the shrimp, people will listen and care about. But again, if I do my traditional science and check, I don't know, growth, reproduction, metabolism studies, nobody will listen to that. Nobody will care about ocean acidification if I tell them if we don't cut carbon dioxide emission, the respiration of shrimp will increase by 27%. That's not a really powerful message. So we decided to focus on something else, which is what people care about and what it is the taste. So we did this experiment where we cultured the shrimp into different pH, simulating today and the future. And then in a random test, we asked people to taste and tell us if they could actually perceive the difference due to the ocean acidification. And the answer is yes. So people could actually taste the difference. They didn't like as much the shrimp raised in ocean acidification condition as compared to the shrimp of today. So that was really exciting because we could make people physically understand what it means to experience ocean acidification. So we could turn this invisible chemical change into something that people could understand. And what we realized through social science studies later is that when people had the opportunity to actually taste the difference, experience physically ocean acidification, they were more willing to change. So people that actually went through this exercise of tasting the shrimp and realizing that has a negative impact on something they care about, these people were actually taking pledges and implementing them in their everyday life. So for example, they were becoming vegetarian, or they were smarter in the way they were traveling, or they were voting for different people. So this kind of science that is more focusing on societal value had the power to drive changes in behavior. So that's the kind of science that we keep doing now. Science that is targeting changes. So science that will actually have a stronger impact on the society. So that's for the mitigation aspect. But there is another angle to this acidification problem, and that's the adaptation. As I said, we need to buy time to resolve the carbon dioxide issue. And to do that, we need to be able to help people locally. So for example, the picture you see here are oyster farmer in Hong Kong. These guys, they don't really care about what's happening globally. What they want to do is keep their job, be able to keep their tradition going, culture oysters in that case, and feed their family. And to help these people, you need local data. And that's where things becomes a little bit more complicated, because you really need to go on the site and understand what's happening to be able to make some kind of projection. What can happen in the future if we do this, this, and this? And ultimately, what we want is maps like that, forecasting what can happen in the future under different scenario of management. What if we reduce pollution? What if we reduce overfishing? What if temperature increase by that level, and so on and so on? But that's a very complicated task to actually create these models, because in every place, conditions are different. So you're going to have multiple stressors, but happening in different conditions. What is a stressor in one place might not be in another one. Temperature might be much more important in a certain place because the organisms are more sensitive. So we end up with this overwhelming task of trying to understand what's going to happen in the future, because you have for every place to understand what are the conditions locally, how they vary, what is actually a stressor, what kind of ecology do you have, the potential for evolution, the sensitivity of the species. So all that seems impossible in a way. It's an impossible task. You can't just do experiment everywhere on everything. So together with a colleague called Hans Bertner, that is one of the lead authors for the IPCC, we thought if we really want to resolve this, we can't just rely on experiments that has to be done. We need to understand the fundamental mechanisms. We need to understand what is happening inside an organism, inside an ecosystem when you expose them to different combination of stressors. So instead of doing what we call stamp collecting, which is repeating the same kind of experiment everywhere, we should spend more time understanding the fine mechanisms. It could be physiology, could be ecology, could be evolution. And that's basically what I started also to do in my lab. We started to understand the mode of action of the different stressors, the performance curve, and we decided to go for sea urchins for multiple reasons. I think it's a keystone species where I live. It's commercially important. And then you have all this knowledge already. We know what's happening to a sea urchin if you change the temperature, decrease the salinity, and so on and so on. But what we didn't know at the time is what's happening inside the sea urchin if you expose them to change in carbonate chemistry related to ocean acidification. So I spent a few years understanding really the fine mechanisms of what's happening inside the sea urchin larvae when you expose them to acidification. And we started with simple things where we collected the eggs, the sperm, built culture, exposed them to different pH. And the first thing we could see, and that's really a kind of a rule for invertebrates, is that if you decrease the pH, they grow a little bit slower. And what you can see on the picture here, they are basically delayed in their development. And then we try to understand the step further. Why are they growing slower? And I'm not going to go into too much detail because I can see that the time is passing and I want to have little time to have questions with you if possible. But basically, we realized that they respirate more, so they basically increase their metabolisms because they are fighting against the stress. And to do that, basically, that's because they need extra energy to cope with this stress that is ocean acidification. And then we had to be really creative. And that's what I really like about my job, is that we have sometimes simple question and you have to be super creative in the way you answer that. And just to illustrate one of those is like we were interested in how acidification was impacting the feeding of the larvae. And these larvae are really small. They are 1/10 of a millimeter. So to do that, we had to basically be creative and use confocal microscopy. And I will just show you the little movie here. But basically, we were filming the larvae under the microscope, and the red stuff are the phytoplankton. And you can see that they are coming close to the mouth here. Then they enter the esophagus. You have a contraction, poof, they enter into the stomach. And then you can basically see digestion in action. So very simple method, just require a good microscope and a good camera and a lot of time. And then what we could do there is basically quantify what was the impact of low pH on their physiology. And I'm going to skip a couple of slides, but basically to cut a long story short, what we could see is that if you expose a larvae to low pH, they need more energy to do the same job. And because of that, at some point, they reach what we call the physiological tipping point. So if you decrease the pH, and that's an example again for sea urchin larvae, from their optimal pH, they grow slower, then they hit the wall, and there's not enough energy for them to just survive, and they go into metabolic depression, they turn abnormal, and they start to crash and to die. So we could use all this knowledge that we developed over the year to build mechanistic models, and now we reached a point where we could actually make projections. So we are able now to build maps like that, where we can imagine what's going to happen under the combination of different drivers and different stressors. And we can use this tool to inform policy and inform managers to ensure that we are keeping the marine ecosystem as resilient as possible to all the horrible changes that we are putting on it. So we can buy the time we need to address the real issue, which is reduce carbon dioxide into the atmosphere. So again, as a young scientist, as you are, I think there's a curiosity aspect in the work we do. But if you're interested in protecting the marine ecosystem, you really have to think about what are my priorities. Is my science designed in a way that it will be able to be used to basically save provide the information to the person that are managing or the policymakers that are there to ensure that there will still be marine life in the future. So what you want to do is to identify the science we need for the ocean we want, which is the motto of the United Nations Decade of Ocean Science that just started a couple of years ago. And if you're interested into joining these initiatives, we just started a program called ORS that has been endorsed by the UN. So it's Ocean Acidification Research for Sustainability. And what we're going to do there is create a roadmap, create basically a strategy on how we can do science to really basically have an impact on society. So cool science, but with a utility to preserve the ocean we want. And I've been really, as I said, my career has been quite fun so far. I've been extremely lucky. I worked hard for sure, but I also been lucky to be at the right place at the right time. And today it took me to these two beautiful places. So I'm sharing my time between Sweden on the top, a small island in the middle of nowhere, and then Monaco, which is the complete opposite. Actually, if I would have taken the picture today, it's completely different. We have 10 centimeters of snow today, so it's really white and pretty. But I also wanted to give back. And as Carla said at the beginning, I spent a big part of my time now trying to share the knowledge we have and share the resources to ensure that everybody can do this kind of research and join the game. And I'm done. So thank you so much for the opportunity, Carla. I know I was quite fast. That's always my problem, I know. But hopefully everybody could pick up a few interesting parts of what I've done and my little story. So thanks a lot. And looking forward for questions if we have time.
Thank you so much, Sam, for a great talk, as always. Yeah, we're used to fast speaking in South Africa, so you don't have to worry about that. So I'm going to use my chair privileges to ask the first question while the questions come in through the chat, I think. So, Sam, you are someone who I believe has really achieved a very good balance between doing your research, teaching, communicating your science, and clearly you're very good at time management. So as you said, this was actually really important for your career development and your opportunities and your luck. So my question is, what advice do you have for the students and early career scientists attending today to achieve a good balance between your career and your life?
That's a tough one, right? Because it's easy for me to say now because as you said, I actually have the chance that I have a family, that I don't think I overwork, but basically I would say work hard at the beginning. That's the difficult truth, is that you have to prove yourself at the beginning and don't get distracted. And that was super hard for me because I wanted all the things I can do now, which is capacity building, communication, and all these things. I wanted to do them from the beginning, but at the beginning you have to be visible. You have to establish yourself. And I don't know anyone in the scientific community that cannot be replaced by 10 other people out there. So we are not special. We are as good as you are. There will be people that are better than you. So what you have to do at the beginning, I would say, is work hard. Focus on the science first. You will have the time in the future to do the rest, and be super social. So that was my success. It's like if you go to conferences, as many as you can. I was always finding a way to sneak in as many conferences as I could just by being, brutal and just writing, say, I would like to come. I don't have any money. Can I come? And sometimes it just works. So travel, meet other people, and don't stay in your room, practice your talk. Go out, go to the pub. Don't be shy. I think most scientists are really nice people. And that's also an invitation here. If you're interested to talk, I think Carla, I'm a quite chill person. So if people want to talk to me or, I'm more than happy to deliver on that. But what made the difference for me was the social aspect, like going out, meeting people, being generous with my time and being focused for sure.
Thanks, Sam. Those are a lot of the lessons that I did learn from you since getting to know you. So before people get upset with me, I've got some chat questions coming in. The first one is from Francesca. She says, while time is running out, what direction would you suggest that researchers in developing countries should take when generally the global damages affect the local challenges? And yet there seem to be not a large voice from the global south. Would stronger North-South partnerships help building some of the knowledge and faith in discoveries and solutions from the South?
That's a really big question. Thank you, Francesca, for that. So the thing is that I've learned recently, I think sure there are a lot of gaps in knowledge in the South for many reasons, but that doesn't mean that we don't know enough to act. So I think the first thing I would say is that we should be less scientific sometimes. That's the thing, is that the policymakers, they don't necessarily want to change, and they use us against ourselves. So basically scientists always want more data. So we need more research, but we don't. I think there are many things we can implement today. So I would say the first advice would be less sciencey and more action oriented, because we know enough. And then for the rest, in the meantime, for sure there should be more partnership with some of the countries in the north, just because they have more resources, and I think it's completely fair. So I think it's important to promote marine research in general and provide jobs and opportunities there. And that's what we try to achieve with the OAICC, where I work in Monaco. So there are a lot of opportunities there. But in terms of priority of science, what needs to be done, I guess today the important part is really to resolve the multiple stressors. And that can be done in collaboration with any countries. There are some theoretical aspects that can be solved everywhere, and then there are some local aspects that can be addressed in some regions. So I know it's not a satisfying answer, because it would take a lot of time to discuss that in detail, but I would say definitely more collaboration everywhere. That's very important. And yeah, that collaboration is the key. I would say that would be my short answer.
Awesome, thank you. And then a question from Nikki James. She said, thank you for the great talk. And did you involve social scientists in your mitigation communication?
Absolutely. So actually we created a center in Gothenburg called the Center for Collective Action Research. And that's basically a very mixed group of people from political scientists, social scientists, psychologists, communicators, and natural scientists. And the shrimp story was really a collaboration between social and natural science, where we first started by sending social scientists in the field to actually investigate about what kind of information were the more likely to drive the change. So I think that's still a weakness in I would say in the global change community world, is that you have these different communities and they are still doing their own things, but their interaction is quite weak. And there are a lot of things that can be done. I really believe that we should start with stakeholder social scientists, and they can tell us what kind of science we need to do to actually be more efficient. And that's exactly what this ORS program that I mentioned is all about. It's about co-design. So we have a work package led by Richard Bellaby and Jesse Turner, where the idea is really to go in the field and talk to people and let them tell us what are the real priorities. And I think that's really exciting. And that's an open invitation. If you want to be involved, just send me an email and I will put you on the list. There will be working groups, there will be meetings and so on. So if you want to get involved, just I can see Rebecca said that she was interested in the chat. So please send me an email and I'll share the list with the leaders.
Thanks, Sam. Yes, and thanks for answering Rebecca's question too. And there are a lot of compliments for you in the chat, so please feel free to stick around. I know you're probably really busy, but if you have time, you're more than welcome to stay and enjoy the rest of the talks from our students. But thank you so much for your time and for speaking to us today. We really appreciate it.
Thank you, Carla. Have a good meeting, guys.
Thanks. So we're a little bit early, so perhaps we should wait a few minutes, but I will be handing over to Tadiwa, who will be taking care of the next student talks. I think the talks are set to start at 2:15, and it's now 9 minutes past on my watch. So perhaps we can just take 3 minutes of a break and then start again at, let's say, 14:13.
Good afternoon, everyone. Welcome to our first session. This is going to be the fish biology and ecology section. It covers a wide range of talks that range from the smallest of organisms, including the parasites, to the fishes, as well as looking at their habitats, their distribution patterns, movement patterns, as well as the impact of some of these environments on commercial line fishing as well. So I hope you're going to enjoy the talks from the different speakers that we have today, bringing interesting talks. So our first speaker is going to be Rudolf Gauss, who's going to be sharing some of the work that he's been doing with fish parasite conservation of 3 endemic rock catfishes, Astrogrannis, from Southern Africa.
Hello, my name is Rudolf, and I will be presenting the fish parasite conservation of 3 endemic rock catfishes, Astrogrannis, from South Africa. The conservation and protection of wildlife is important and necessary to maintain healthy and functional ecosystems for the future. Moreover, approximately 40% of all animal species are parasites, and less than 10% have been described adequately. At species level, parasites benefit the host by enabling them to survive. They also play an important role in managing ecosystems in which the hosts live. In some scenarios, parasites can assist in species control within ecosystems where competition and predation do not control natural populations and can prevent monoculture. Compared to global parasite diversity, information on South Africa's parasitic species is scarce. South Africa harbors a highly diverse freshwater fish community with 45 endemic species that are recognized as endangered on the IUCN's Red Data list. 28 of these are endemic to the Cape Fold region and classified as threatened. The current project aims to lay the foundation for parasite conservation by linking the host conservation status as well as the biodiversity conservation through identification of generalist and specialist parasitic species associated with fish hosts. The unique catfishes belonging to the Astrogrannidae will be used as the model to identify associated parasite species that may be of conservation concern or assist in the conservation of these endemic catfishes. These species are used because nobody has worked on them before and no parasitic community data is available for these 3 species in the family with Astrogrannis barnardi and Astrogrannis guinea listed as endangered and near threatened, respectively, while the conservation status of Astrogrannis clatteri is of least concern. All 3 species have no records of parasites. These 3 species are all in different conservation categories. Astrogrannis barnardi and Astrogrannis guinea are confined to the tributaries of the Clan William Olifant system in the Western Cape. Astrogrannis barnardi and Astrogrannis guinea can be found in cobbles and clear flowing waters. Astrogrannis clatteri is confined to the Orangeval and Olifant Limpopo system of South Africa. They are found in rocky habitats with flowing waters. A total of 15 individuals of Astrogrannis clatteri was collected from the Fall River at the Kwasa Youth Camp, and 15 individuals was collected at Skuman's Drift Bridge. Prior to the collection of the fish and commencing of research, permits were obtained from the relevant permitting officers and ethics approval from the NW Animal Care Committee to utilize animals for research purposes. The specimens collected at the Kwasa Youth Camp was small, and those collected at Skuman's Drift Bridge was large individuals. The importance of collecting big and small species are that they have different preferred diets and may have different parasites. According to data from Truther, it shows that a minimum of 15 fish per species per site is needed to determine a full biodiversity report of the parasite. This means that we don't need to collect more than 15 fish per species, but if we collect less, we might not get a full biodiversity report. Fish were collected using an electroshocker. The fish were placed into aerated containers with water from the rivers the fish were collected from and kept alive for transport to the Northwest University. Water and sediment was collected at both sites. The fish were identified using a guide of the freshwater fishes of Southern Africa. The fish that were collected were mainly killed by severing their spinal cord and piffing. All individuals were screened for external parasites on the body surface and fins, and the blood sample was collected to screen for blood parasites. Fish were measured, a total, standard, and fork length was taken. The fish was weighed and the photograph was taken. The gills and fins were removed and placed in water. The intestine, stomach, gallbladder, liver, eyes, and brain were removed and placed into saline where the organs were fully examined for any endoparasites under a stereo microscope. Muscle samples were taken for metal analysis from the specimens. Parasites that were observed were collected. The best way to fix nematodes is to use hot 4% formalin. Scanning electron microscopy was used for morphological characterization and studying of relevant taxonomical structures. Line drawings for the parasites was made using a compound microscope. A dope illustrator and a Wacom drawing tablet was used for ink drawings of parasites. Literature study that was used to identify the species was Moravec 2010. DNA extraction and subsequent PCR amplification of selected genes were performed using the nucleus-spin DNA kit and the Proflex thermal cycling machine, respectively. Positive PCR products were purified and sequenced by Inquaba Biotechnical Industries, and the obtained sequence were inspected and trimmed using the ingenious software package. Ribosomal genes such as the 20S rRNA region and the Cox barcoding gene region will be included in phylogenetic analysis from comparison to closely related species. A total of 30 Australianus clatteri was collected at both sites. The rock catfish at the Fall River were only affected by 2 species of nematode that was found in the intestine and body cavity of the fish. Both species have only been identified to genus Raptogona and Estrongylidus. Only female Raptogona species were observed. Nematodes are roundworms and are among the most abundant animals on Earth. They are parasites that occur in animals and can be found in marine and freshwater environments. These nematodes was found in the intestine of the Australianus clatteri. Overall prevalence was 38.71%, and 2 nematode species, Raptogona was 35.48% and Estrongylidus was 6.45%. And intensity of infection ranged between 1 and 17 and 1 for the 2 species, respectively. The Raptogona species that was found have not yet been identified to species level, and with morphological characterization, it is possible that a new species have been found. Unfortunately, no male representatives were found. This nematode correlates the most with the genus Raptogona as seen in the picture diagram. This species was placed in the genus Raptogona with the placement of their stoma and vestibule as well as the apical view. Estrongylidus have been identified by the characteristics of their anterior and posterior ends, as seen in the diagram. Estrongylidus are parasites that have birds as final hosts, and only 3 species are known in the genus, and this genus are known to have zoonotic potential. Future work for this project includes sampling in the Western Cape for Australianus barnardi and Australianus gilli. The genetics and morphology needs to be finalized for the parasite specimens that have already been found and the new ones that may be found in the other 2 target fish species. I would like to thank the following individuals for their assistance in the field as well as with lab work. The Northwest University Water Research Group is thanked for providing infrastructure and facilities for sample collection and processing. I would also like to thank the Northwest Permit Office and Cape Nature for allowing me to collect my target fish species.
Thank you very much for the interesting talk on the parasites. Remember, if you have any questions, please post them in the chat so that we can ask the speakers. And I would like to also remind you to pay attention to the talks and place votes for the best MSc and PhD talks at the end of the sessions. So we have a bit of time until the next talk is supposed to start. So I'll start off with maybe one question. Okay, Rudolph, my question is, did you notice any differences between seasons, between the different type of parasites that you were finding, or were you only able to sample once off?
So we've only been able to sample once off because we have only a limited number of specimens and like a limited number of time we can sample our specimens. But so far we've only sampled, or we've now just finished sampling the Western Cape specimens as well. But we're not sure yet what differences are in parasites.
Okay, thank you. We still have some time for more questions if anyone wants to ask questions. Okay, I'll ask another question. You mentioned that the intermediate hosts, because you spoke about some of the intermediate hosts being birds. Do you have any idea of which birds maybe in the intermediate source for this new species, the one that you thought might be a new species?
So with the Estrongylidus or the intermediate dose for birds, but we have not yet identified it till species level. So we're not sure what species the parasite is. So with that, we're not sure what bird the intermediate or what intermediate dose it is.
Okay. Okay, so we have a question by Albert in the chat, and his question is, considering the threat status of some of the study species, are there any possibilities for non-invasive collection assessment for parasites, or any recommendations, e.g. the possibility of eDNA?
So I'm not entirely sure yet, so I'll have to come back to Albert with that question.
Okay. So are you also are you collecting the DNA specimens because even if you do go for the eDNA approach, you probably going to need a reference library. So in your collections, do you also, because I saw you mentioned fixing them in formalin, do you also keep any of the specimens for DNA work later on, maybe at another stage?
Yeah, so all the specimens that we collect are I think that we need to, or we want to create genetic barcodes for all the specimens we collect.
Okay, thank you. Okay, I think we can now move on to our next speaker, was Mihle Gaiza. He is going to be sharing his talk on the necessary provision of macroalgae use in the coastal seascape of Algoa Bay. Thank you.
Good morning, I am Tembelihle Dube, and my presentation is titled Building the Foundation for Efficient Monitoring of Mesophotic Fishes to Support Management of the Tugela Banks Marine Protected Area. Global fisheries statistics from the 1950s to present day indicates that fish stocks are in decline both locally and globally, and the anthropogenic activities such as pollution, human-induced climate change, and overexploitation are the cause of the decline in fish stocks and therefore pose threats to marine biodiversity and the essential services provided to humans. Marine biodiversity, particularly fish populations, are very important to humans for their survival and well-being. We as humans depend on fish as our source of protein that is easily available to us and is rich in omega-3 fatty acids. Fisheries provide food for billions and livelihood for millions of people worldwide, and fish populations contribute to economy through fishing and thereby creating employment for fishermen and those who operate in the entire chain from construction vessels and equipment supplies. Considering that fish is an ecosystem product, it is therefore important that a well-functioning marine ecosystem will create the environment that is good for fish populations to inhabit or to thrive in that good environment. And also healthy marine ecosystems act as tourist attractions. For example, coral reefs do attract divers, and it has been noted that tourism is known to have more profits compared to fishing. Fish populations have been managed using traditional management, which is the single species management that is only based on managing a single species and does not include the ecological relationships between the species as well as the environment. It includes catch limits, size limits, and equipment restrictions, for example, long line and spear fishing. These equipment restrictions help to protect the non-targeted species and also ensure sustainable fishing of the targeted species and reduce bycatch as well as habitat alteration. The single species management is not effective as it is not holistic and it is very expensive. Another management approach is an ecosystem approach to fisheries, which is an integrated management approach that promotes conservation as well as use of the whole ecosystem. It is applied for managing fish populations, and one example of the ecosystem approach to fisheries management is marine protected areas, which have been implemented to better manage anthropogenic impact and also protect marine biodiversity. Encouragingly, there's a growing literature demonstrating that MPAs can play an important role in marine conservation if appropriately managed, and also MPAs can be utilized in order to supplement the traditional fisheries management. And not only do they offer a way to promote long-term economic viability and sustainable fisheries, but they promote chances for marine research and education while supporting the preservation of habitats as well as unexploited species. The Otuela Banks Marine Protected Area is a newly established MPA created in 2019. It is in KwaZulu-Natal province, and to date there's been only limited research and monitoring. That is why the study is aimed at assessing the patterns and the demersal fish assemblages and how the environmental variables influence the structure of demersal fish assemblages and fish diversity. It is also aimed at providing a baseline assessment of the status of fish populations and the different management zones of the MPA, and also develop a feasible long-term monitoring program that can be implemented in order to support efficient management of the MPA. The study was conducted on the shelf of Central KwaZulu-Natal in the Otuela MPA, which is located between St. Lucia and Durban. This MPA has a total size of 4,094 square kilometers. The map shows different zones of the MPA, which are the controlled pelagic with list as well as restricted zone. It also shows mapped sites nested within the management zones, as well as 9 sites that were sampled and had 14 stereograph replicates. To achieve my objectives, key reef sites in the different zones of the MPA were identified and mapped with a multi-beam echo sounder. The mapping information was used to design a stratified random and a spatial balanced sampling protocol for baited remote underwater stereo video system surveys using QGIS to interpolate depth, slope, or relief. Baited remote underwater stereo video systems, which are stereographs, are a proposed non-extractive tool employed as a long-term monitoring design that helps to estimate size distribution and relative abundance of fish and reef species to a wide depth range. Stereographs create a permanent record that can be reviewed in order to reduce inter-observer variability. The system shows 2 high-definition cameras and a bait canister that is used to attract fish, and it is connected to a bait arm. And then we have the rope that is connecting to the buoys. The stereograph system was calibrated before and after sampling in order to allow for accurate length measurements of fish appearing in the field of view of the camera, and the system was deployed to record on a seafloor for a duration of 60 minutes. The pictures here show the fish assemblages that are observed using stereographs. Videos obtained from stereographs are being analyzed using event measure software to obtain data about diversity, abundance, and size of the fish. Transect measure software will be used to measure habitat complexity. This is the interface for transit measure software, and this is the interface for event measure software. General additive models will be run to determine the effects of environmental variables and spatial variables on the univariate matrix. And then permutational multivariate analysis of variant will be connected to assess the patterns in fish assemblage structure. To design the long-term monitoring program, species accumulation curves, power analysis, and cost-effective analysis will be run. The data will then be analyzed in Prime V7 and in R via RStudio interface. The outcome of the study is based on research, which is essential as it provides starting point to track changes and inform the design of future monitoring and management efforts. The knowledge on species distribution can assist managers make decisions about which species to protect. In order to ensure effective management of marine resources, critical habitats supporting species that are ecologically and economically important need to be identified and protected throughout their life cycles. It is also necessary to keep track of changes over time and signal which strategies need to be revised based on the observed trends in the data. For my progress, I've managed to complete literature review, data collection that was successfully carried out in June 2022, and a total of 131 samples that were collected from 9 websites in the offshore management zones in the depth range of 40 to 190 meters of the Otwela Banks, MPA. I'm currently analyzing those videos. I still need to do species measurements and habitat classification, data analysis, as well as write-up. These are the sources that I used to compile the presentation. I would like to acknowledge these organizations for funding this research and making it possible. Thank you so much for listening.
Thank you very much, Tembelihle, for that presentation. I'm sorry I made the mistake. I introduced the wrong speaker at the beginning, but that was a talk by Tembelihle. And any questions for her? I can see a couple in the chat. So for Tembelihle, the first question is, hi Tembelihle, it's for great talk. I am also using browse for my MSc project and have identified other species with other visual techniques other than browse. So I want to know if you think the stereo browse are effective in providing a true representation of species richness and total abundance in the Utsekela MPA.
Hi everyone, thank you Tembelihle for the question. So I can say that yes, stereographs are being effective in representing the species, the representative species, because they've been used globally to study fish assemblages. However, there are some biases which are being accounted for, considering that we're using the standardized protocol. Yes, for instance, since we're using baits, we do account for that in covariates such as visibility, temperature, and other covariates. Yes. And also, since we're using baits, it does help to increase the relative abundance as well as diversity of the fish species that are being targeted by the fisheries. And that does not prevent the sampling of other fishes that are not attracted to baits. I think that answers your question.
Okay, thank you very much for that answer. Another question coming from Nobuhle. She says, nice study, Tembelihle. I just wanted to ask if there's a reason why, a specific reason why you've only focused on mesophotic reefs and excluded inshore areas.
So the Otwela Banks Marine Protected Area is a newly established area, and few studies are focused on offshore areas, with most studies focusing on the inshore areas. So the reason why we targeted mesophotic reefs, it's because it is like from the different studies, it does show that more fisheries is taking place in the mesophotic reefs, and also the Utwila Marine Protected Area. The reason why it was established, it was in order to protect the mesophotic reefs. So that's one of the reasons why we targeting them, and also considering that the Utwila MPA is close to it. There's a closed what do we call this? Also close to it there is a big river mouth, and therefore meaning that considering that we're using the stereographs, which are a visual technique, it won't be easy to get the visual samples. So yeah, that's why we targeting the offshore areas where it's clear, like considering that we're using graphs, so we're able to see the different fish or different demersal fishes appearing in the field of view of the cameras. I think that answers the question too.
Okay, thank you very much for that answer. We no longer have time for questions. We can move on to the next speaker who I've already introduced. So we are going to have the next talk by Michael Gaiza. Thank you.
Good day. My name is Mitle Gaiza, and I'll be presenting my master's research titled The Importance of Structural Complexity for Necessary Provision Within a Mosaic of Habitats in a Temperate Rocky Cove. I'm supervised by Professor Nicola James, Dr. Paul Pierre-Staines, and Dr. Anthony Bennett. Coastal marine and estuarine ecosystems are highly productive and serve an important necessary role for coastal fish. Most research into the necessary function of coastal ecosystems has been focused on the estuarine environment, particularly in South Africa. The value of estuaries as necessary has been attributed to the provision of structurally complex habitats such as seagrass, mangrove, and salt marsh, which results in increased survival of fish and invertebrates through the protection from predators and food provision. Early studies inferred the necessary role of estuaries by the density of juvenile fish. Beck et al. 2001 later argued that habitats within estuaries need to be considered separately, with the necessary role of a habitat based on the proportion of juveniles moving to adult habitats per unit juvenile area, which is greater in necessary habitats. The greater migration to the adult population in necessary habitats is through a combination of higher juvenile densities, growth, and lower mortality rates. In nearshore habitats without seagrass and mangroves, other ecological complex habitats such as reefs dominated by algae may provide important necessary areas for juvenile fish, particularly in sheltered bays where wave action is reduced. Which brings us to the aim of this project, which is to assess and map habitat complexity and macroalgae as well as fish assemblages across 3 different habitat patches in a shallow sheltered rocky cove in the western sector of Algoa Bay. The main objective is to assess the necessary function of habitat patches of differing complexity within the cove for juvenile fish. The study site for this project is Flat Rocks in Algoa Bay, situated in Gqeberha, formerly known as Port Elizabeth. It is on the southeast coast of South Africa. It is sheltered from the dominant westerly winds and southwesterly winds which are found in Gqeberha. There are several communities of macroalgae such as upright coralline algae and fleshy seaweeds. The habitats were defined into 3 habitats: 1, sandy patch; 2, low-profile reefs dominated by Lawrencia species; 3, high-profile reefs dominated by Leucemium species. Gratwica and Spite 2005 designed and used the Habitat Assessment Score to assess habitat complexity across different shallow marine habitats, including algal beds, sandy patches, and seagrass beds and reefs. It measured rugosity, variety of growth forms, height, refuge size categories, percentage live cover, and percentage of hard substratum. I used the same habitat assessment score to define the level of complexity of my different habitats that are found within my study site. Rugosity was also measured using a tape and chain method, whereby a 2-meter chain was deployed on the substrate and measured. The measurement of the deployed chain was deployed by the original chain length, and the closer to 1, the less rugose the habitat was, and the further away from 1, the more rugose the habitat was. Fish assemblages as well as fish behavior were observed using remote underwater video systems, which were also deployed in the different habitats and observed using event measure. High-profile reefs, which had an increased variety of growth forms, height, as well as macroalgae species that were branching out, scored the highest, meaning that they were the most complex according to the habitat assessment score. Low-profile reefs scored second highest, and the least, which were pretty much bare with little or no growth form, were sandy patches. The rugosity index from the chain and tape method showed us that high-profile reefs were the most rugose habitats, followed by low-profile reefs, and the least rugose were sandy patches. Further results of this study also show that there was a greater number of juvenile fish that were observed compared to the number of adults. The proportion of adults observed was greatest in sand patches and lowest in high-profile reefs. The mean MXN was highest in high-profile reefs and least in sand patches. The Shannon Diversity Index, which are basically measured the diversity of fish species that were observed, was lowest for high-profile reefs, meaning that there was least diversity observed in high-profile reefs, and the most diversity was observed in low-profile reefs. The mean number of species was lowest in high-profile reefs, and it was highest for low-profile reef. An NMDS plot based on the breakages similarity was ran to see the correlation in fish assemblages between the different habitats, and the NMDS showed that there is no correlation between the fish assemblages that were observed in the different habitats as seen in the images in the NMDS provided. And also the presence and absence was also ran, and it showed a similar trend. Sappa salpa, Diplodus cavernus, and Diplodus capensis were the three fish species that displayed the observed behavior the most. And for this presentation, I am highlighting on them. The wandering behavior, which was behavior of a fish swimming within the field of view for more than 30 seconds, was mostly observed in high-profile reefs and the least in sand profiles. The transitory behavior, it was when a fish swam within the field of view or within the sight of the habitat for less than 30 seconds and then left the field of view. The feeding behavior, which was also mostly observed in high profile and least in sand patches, was when a fish demonstrated a feeding behavior, which was scraping the subscrape or scraping the macroalgae present on field of view. For this study, I conclude that habitat complexity had no effect on fish assemblages and fish abundance. It did, however, have an effect on juvenile fish behavior, in particular feeding and wandering behavior, which were mostly observed in complex habitats. The feeding, wandering behaviors, and high abundance of juvenile fish presence suggest that the area does provide necessary provision. The consistency in fish assemblages between the different habitats might suggest that the habitats are connected through habitat utilization by similar species. Thank you very much for listening, and if you do have questions, please do ask, and please don't forget to like and comment and follow us on our Instagram page as well as on our Twitter page. Thank you.
Well, thank you very much, Michelle, for an interesting talk. I think we have time for a question or two. So the first question comes from Francesca, and she's asking, "Hi, Michele, interesting study, and it must have been challenging to take measurements in such a dynamic environment. Did you do any validation on the possible inflation of flow rugosity versus high rugosity in sand and algal canopied quadrants to fully optimize the methodology?" What I mean is that to take a flat measurement might have been easier than on convoluted algal canopies while underwater and wave action.
Good day. Thank you very much, Tadiwa, and thank you very much, Francisco, for the question. In terms of validation, I'm not sure, but in terms of rugosity, we measured it two ways. Visually using the habitat assessment score method, which is suggested by Kratwika and Spite 2005, and then we physically measured it using the chain and length method, which pretty much gave us similar results. And then there was a study as well which compared visual assessments of rugosity as well as physically measuring it. And the study found that visual assessments as well as chain and tape method, they do pretty much give similar results. Yeah, thank you.
Okay, thank you. We have one more question in the chat, but only just like half a minute. So I kindly ask that you may please respond to this question in the chat. In the meantime, I think we can move on to the next speaker, Andrew Michael-John, and he's presenting on the spatial and temporal analysis of fish assemblages within the Neisner estuary system.
Good afternoon. This afternoon, I'm going to be presenting to you one component of my master's thesis titled Spatial and Temporal Analysis of Fish Assemblages within the Neisner Estuary System. The components I'm going to be presenting to you today is the distribution of various estuarine gills within the Neisner Estuary. The Neisner Estuary has been given the highest conservation priority of South African estuary. This classification was based on weighted size, habitat, zonal type rarity, and biodiversity in the system. It's a unique system with a large marine bay and is classified as an estuarine bay system. In the Neisner Estuary, up to 200 fish species have been recorded, and this system has an open mouth with a very deep channel, which has led to a higher proportion of marine fish present in the system than other estuarine systems similar to it. The Neisner Estuary can be broken up into 4 distinct regions. These regions are the Marine Bay, which is closest to the mouth. This is what makes the Neisner system unique from other estuaries on the southern coast of South Africa, the lagoon, the lower estuary, and the upper estuary. For this work, we are going to be using spatial mapping and use the spatial mapping to guide conservation. By using spatial planning, it allows for a more targeted approach to conservation. Spatial conservation in Neisner Estuary is currently made up of various sensitivity zones. These sensitivity zones are, however, not guided by fish distribution, but rather guided by habitats within the system. The sampling methods used for this master's thesis, we used 2 different types of sampling methods. The beam trawl sampling, which was made up of 2 sampling trips. These beam trawls are ideal for targeting demersal habitats, and it makes use of a 1.5 meter net with a 0.5 meter net mouth, which is trawled at a speed of 2 knots for 5 minutes where possible. In certain sites with heavy vegetation, this was not possible, and the trawls were therefore shortened. The beam trawl was made up of 24 different sample sites, all of which were within the vicinity of SAIAB sample sites where possible. The SAIAB sampling was also made up of 2 sampling trips. The SAIAB is ideal for targeting littoral habitats, which means that we are able to target both the demersal and the littoral habitats, which has not been done before in the Neisner estuary, allowing for a better idea of all the fish species within the system. This made use, the SAIAB used was a 30-meter SAIAB and 30 sample sites were used, although for this analysis of estuarine gills, there were 4 additional sites in the Thiessen Island canals, which were not included in this analysis. So 26 sites made up the estuarine classification analysis. For this analysis, all the fish species caught were split into their different estuarine guilds as classified by Whitfield 2019. These are on display at the moment. The total catch for these sample periods were made up, the beam trawl was 2,894 different individuals making up 24 species, and the SAIAB was 26,373 individuals making up 42 species. For each of the different sampling methods, the mean density of catch from each site was calculated for the 2 sampling trips. These results were then transformed. A square root transformation was used for the beam trawl catches where there wasn't such a great variety in catch numbers, while a fourth root transformation was used for the SAIAB catches where there was a much greater variety in catch numbers, and the kernel densities from these results were plotted using ArcGIS Pro. For all of the different beam trawl guilds, it appeared as though the hotspot was found in the Marine Bay area of the system, with marine estuarine dependence showing a secondary hotspot just at the start of the lagoon section of the system. SAIAB species tended to show wider variety in their hotspots, with estuarine and freshwater, marine and estuarine and freshwater opportunists showing extended hotspots through both the Marine Bay and lagoon stretches of the system, with only the solely estuarine and marine estuarine dependence showing hotspots in the Marine Bay, whereas marine stragglers had such low catch numbers with only 2 individuals sampled, so it's difficult to gauge hotspots from that. Of all the habitats within the system, there seems to be 2 major vegetation groups used by species. The first is Zostra, which shows widespread use by all estuarine guilds, with the majority of solely estuarine species being present in areas of Zostra. Calerpula is the second most abundant, and it's less common in the system than Zostra, and also slightly less productive in Neisner than Zostra, showing lower catch densities and less variety of catch. The third major habitat type is estuarine open water, which shows the lowest catch density as it provides very little protection for juveniles. In the Neisner estuary, this tends to be areas close to deep channels. The output from this project shows that although the habitat-specific sensitivity zones is the easiest way to spatially manage a system, it does lack sensitivity for areas around important habitats. Lots of fish species are using the complex interconnected habitat zones to move in and out of, and are therefore not sitting in a certain type of habitat. So it's important also to conserve these areas between important habitats. And I've highlighted on the map there one specific area which sits around a whole lot of Zostra beds, and we got very high catch densities in this area, but it falls under a low sensitivity, meaning that the fish are not protected appropriately in this important habitat. So thank you to all of you for listening, and I'd just like to say a big thanks to my supervisor team and also my funders, in Neisner, the Neisner Basin Project, and Thiessen Island Homeowners Association. Any questions are now welcome.
Thank you, Andrew, for the interesting talk. I remind you guys to please ask questions in the chat box and to interact even after the speakers already presented so that we can just continue interacting. So there's a question for you from Francesca within the chat, and she says, thanks a lot, Andrew. Do you see any possible role for coastal development on your results? Any small barriers?
So yeah, in Neisner, the one big coastal development that we were working with was Thiessen Island, and I had additional, as I said in the introduction, I had additional sites in Thiessen Island. And from my further analysis, it appears as though not that many fish are making use of this system there. So in the further analysis, we had identified key species, and it was only the mullet species that are making use of that coastal development. But where that coastal development seems to be quite important is for your flagship species, the Neisner seahorse. They seem to be making quite wide use of that. We got most of our samples of Neisner seahorse in this area, and it's well known that they use this area. So although it doesn't seem to play a major role for the overall estuarine ecology, it appears as though certain species are making use of this.
Okay, thank you. There is another question by Albert. Out of curiosity, what freshwater species did you collect?
So in Neisner, we got very, very few freshwater species. I think in my sample, it was only estuarine and freshwater species, and I would need to look at my results, but I think it's just one, just Glossogobius caladus, which I think that was the one that was an estuarine and freshwater species. Neisner's salinity almost to the end of to where we stopped working, which I think is about 15 k's up from the mouth, is still, your salinity is still about 27 parts per thousand. So it's very, very marine dominated.
Okay. Just quickly, there's a question, I think, follow-up from Francesca, and she says there may be some urban tolerance species locally. I think it's a follow-up to the previous question. So I don't know if you want to respond to that quickly.
It was just
Yeah, so sorry?
Okay.
Just a comment more than a question. Thanks.
Okay.
Thank you.
Yeah.
Okay, if you can answer the last one in like a minute or so. Okay, so hi, Andrew. I noticed your project also has temporal aspects. So have you been seeing any seasonal changes in your catches?
Yeah, so in terms of the temporal aspects, that was mostly to do with the seine net sampling, because we did a winter and a summer sample set for seine net. And definitely my summer sample set, I got much higher catches. And also an interesting thing that I'm starting now analyzing the results further, we're starting to see is it looks like thermal refuge may have played a role in some fishes' distributions, because areas with the highest temperature showed widespread catches during a time where it looks like there was upwelling in the system. So that it looks like during summer, marine fish are making use of the system as a thermal refuge. And it was especially interesting because we caught a lot of shad in this area, which is quite an important fishery species.
Okay. Thank you very much. Please keep the discussions going in the chat. It's very interesting, and we look forward to your responses to some of the questions. So now we can move on to thank you. So now we can move on to the next speaker, Timbani Mkize, and they're presenting on the title, Habitat Use by Juvenile Coastal Fish in Vegetated Habitats of Algoa Bay's Shallow Water Seascape.
Greetings, everyone. My name is Timbani, and I'll be presenting my master's results. Different habitat types within a broad and necessary seascape have different structural complexities, and they play different but important necessary roles. Habitats with higher structural complexity are important core necessary habitats as they support high abundances and richness of juvenile fish through both the provision of protection and substrate for food to grow. The majority of studies on nursery function focused on seagrass due to higher juvenile densities in seagrass compared to other habitats. Very less attention has been paid to habitats like macroalgae. The disparity between the number of studies assessing the nursery function of seagrass and macroalgae may be attributed to the difficulty of using traditional net sampling techniques over rocky bottoms that support macroalgae. So remote underwater video systems overcome this sampling limitation and can be used to study fish assemblages in habitats that are difficult to sample using traditional sampling techniques. The aim of this study is to assess the necessary role of seagrass and red macroalgae for juvenile fish in the Alcoa Bay shallow water subtidal seascape using ministerial rafts. The first objective of this study is to assess the efficiency of ministerial rafts, collect video imagery data that allow for the measurement of 0+ juvenile fish in the shallow water habitats. The second objective is to assess habitat connectivity and habitat use by juvenile fish. The two habitats of interest were situated in the lower reaches of slopes and flat rocks. So our ministerial raft systems were developed following the system made by SAIAB. I'm not going to go into details in everything, but the most important part here is the distance between the camera lenses to make sure that smaller juveniles are showing and can be measured. The distance had to be kept at 200 millimeters. This is our ministerial RAF system that we developed. As you can see on here, the distance between the two camera lenses is 200 millimeters. You can also see the height of the system, which is 450 millimeters. And then on B, I'm showing the painted system, which was painted because the unpainted one had reflections that chased fish away. Tests were conducted to determine the length measurement accuracy, the number of deployments needed in each sampling site, and the duration of deployment. The systems were calibrated using CGIS calibration software and a calibration view. When it comes to data collection, deployments were done simultaneously in 3 sites per habitat type. Deployments in seagrass sites started 20 minutes before high tide to ensure that sampling was on the slack tide for optimal water clarity and full submersion of seagrass patches. Deployments in macroalgae sites were conducted 20 minutes before low tide to ensure that sampling was during slack tide for optimal water clarity and easy deployment and retrieval of the ministerial rafts in the nearshore marine environment. Ministerial rafts were left to record for an hour before they were retrieved. Collected videos were analyzed using event mesh with fish identified to the lowest taxonomic level possible. Max N was recorded. Total length was also recorded using CGIS stereo lab. The behavior of individual fish was analyzed using Boris. Data was analyzed using Statistica and Tribe. Before I get into the results, this is just to show you the structural differences between the two plants. Cumulative number of species increased with time, but the rate was higher for macroalgae compared to seagrass. The increase stops at minute 45, which means 45 minutes can be used as the minimum deployment time. When it comes to the number of deployments. Cumulative species number increased with the number of deployments, but for macroalgae, it stopped after 5 deployments, whereas for seagrass, it stopped after 2 deployments. But we used 5 deployments for both habitats. We managed to measure the smallest individual, which was 15 millimeter. A total of 24 tags were recorded in both habitats combined. At least 18 tags from 10 families are recorded in the macroalgae, and at least 11 tags from 6 families in the seagrass habitat. These 2 habitats were massively dominated by berries. In terms of the number of species, macroalgae had a significantly higher number of species compared to seagrass. As you can see on graph A, this is also showing on the Shannon diversity index graph on graph B. And then in terms of abundances, there was no significant difference in the total mean abundance between the 2 habitats, even though it was slightly higher in the seagrass compared to macroalgae. And the macroalgae habitat in terms of abundances was dominated by striped and black-tailed. But when it comes to seagrass, the most dominant species were the Ape-stump nose and striping. The mean total length of fish between the 2 habitats was not significantly different, but it was slightly higher on macroalgae compared to seagrass. And when it comes to percentages, juveniles were comprising of over 70% of species recorded in both habitats. About 6 tags are common between the 2 habitats. And those include the stripy and black-tailed that were significantly higher in macroalgae compared to seagrass, but they also include the Ape-stump nose that was significantly higher in seagrass compared to macroalgae. When it comes to total assemblages, the juvenile fish assemblage differed significantly between the 2 vegetated habitats, while there was no variation between the sites within each habitat. Similarity percentage analysis shows there is a 57% similarity between the 2 habitats. Table 1 is showing species that are contributing the most to this similarity. In terms of behavior, there was no significant difference between the 2 habitats. And when compared between the 2 habitats, all behavior categories showed no significant difference. Just to shed light on fish behavior and habitat use, slow mineral behavior and feeding seem to be dominating both habitats, and it's important to note that these behavior categories are associated with higher habitat usage by fish. In conclusion, both habitats are important as fish nursery habitats. Both habitats were dominated by juvenile fish, with juveniles showing behavior indicative of habitat use, which is highlighting their necessary role.
Thank you very much for that talk, Temani. The next, we have a few questions in the chat. First one from Francesca, and she's asking that even if the study seems largely aiming to optimizing the camera design, do you think that the 2 areas chosen for the Zostera and may be limited, potentially making the data from the replicates of the habitats not independent, i.e. maybe similar assemblages were recorded among the 3 replicates within each habitat because replicates were also chosen from the same area within one habitat.
Thank you, Francesca, for the question. Yeah, the three sites were actually chosen in the same habitat, and I think that had a lot of impact when it comes to the comparison between sites. But we largely focused on comparing seagrass and macroalgae, how the fish use those 2 habitats, because it was already known that seagrass is mostly important as the nursery area. So we tried to actually compare it to macroalgae and find out if macroalgae is playing the same role as seagrass. But when it comes to the sites within the habitat, yeah, there might be a lot of bias because they were very close to each other.
Okay, thank you very much for that answer. I'm sure I can reply to any other further questions within the chat. For now, we can move on to the next speaker, Dinam Kari, and she'll be speaking on the movement, behavior, and migration of catadromous fish species, Anguilla mozambica, Pseudomyxus capensis in the Kari River and in South Africa.
Hi, my name is Dinam Kari, and I'm a first-year PhD student at Rhodes University based at SAIAB, and I'll be presenting my PhD proposal. The title of my presentation is Movement, Behavior, and Migration of 2 catadromous fish species in the Kawi River system of South Africa. In decades are seeing a decrease in aquatic biodiversity, and this is attributed to anthropogenic activities such as habitat alterations. Some of the species that are highly impacted by this are migratory fish species, which need to undertake vertical migration up and downstream rivers, estuaries, and oceans as part of their life cycle. Possible barriers along the way include habitat degradation, water diversion, and damming, which lead to loss of connectivity between the 3 environments. Fish migrate for multiple reasons, can be food availability, environmental condition, and some migrate as part of their spawning cycle. The fish that migrate from the sea to freshwater to reproduce are called anadromous, and the ones that migrate from the freshwater to the sea are called catadromous. The 2 species that will be used for the study are both catadromous species, and I have included a diagram here showing an example of their life cycle. Just to give a little bit of background about my study species, the first one is the long-fin eel, Anguilla mozambica. We have about 19 species of ungulate eels globally, and 5 of them occur along the coast of Africa. This is one of them. It has been shown that eels contribute towards food security and livelihoods, and in some regions, they are known for their spiritual significance. They are top predators which live all the way up to 15 years, reaching maturity between the ages of 8 and 10, with females reaching maturity earlier than males. Literature shows that their patterns are mostly residents in the freshwater environments, but some of them show habitat shifting behavior. Eels have recently been updated on the IUCN Red List to near threatened due to their decrease in abundance quite recently. My second species is the freshwater mullet, Pseudomyxus capensis. Mullets are known to be quite popular in South African estuaries and freshwaters. They are well known in aquaculture, commercial, and subsistence fisheries, and some people use mullet as bait to catch bigger fish. They are primary consumers. They live up to 12 years and matures earlier than eels, which is about the ages of 2 to 3 years. and their recruitment in South Africa has been shown to occur between September and November. And in some cases, they are more abundant in estuary reaches compared to rivers. Both of the species are catadromous, and spawning is their most susceptible stage to human activities and barriers. And therefore, understanding both biotic and abiotic interactions associated with migration of these two species is important to develop species-specific conservation plans for both of them. There is limited information on mallets, especially the life cycle and movement of mallets in South Africa, more specifically the freshwater mallet. However, there is information available on the life cycle and migration of eels. Work that has been done on eels migration provided evidence that they spawn in the Indian Ocean in an area called the Mascarene Plateau, which is on the eastern side of Madagascar. After hatching, the larvae migrate towards the continental shelf, mostly as a result of the Agulhas current, up the river mouth and into the upper reaches of the rivers as a yellow eel stage. This is where it will stay until it reaches reproduction maturity and then migrates back into the sea. There is no data available about the movement and habitat use of the yellow eel stage of eels in freshwater, especially these eels, and then the frequency and duration of their movement between the estuary and the freshwater before they migrate back into the saltwater, and if there's any environmental conditions or factors that influence their migration, which leads me to my aim and objective. Therefore, this study aims to use acoustic telemetry to study the movement and migration patterns of two catadromous species in the Kawi River system in the Eastern Cape province of South Africa. So this is the freshwater mallet and the longfin eel. So the first objective will be to study the frequency and duration of the movement of these two species between freshwater, estuary, and the coastal habitat, and to link their movement to environmental parameters and geophysical cycles. And furthermore, we will link the seasonal movement of freshwater mallet to their spawning behavior. This study will take place at Kawi River, which is located in the Eastern Cape province of South Africa. The river flows into the Kawi estuary, which is 20 kilometers, and drains into the Indian Ocean. SAIAB has 20 existing reservoirs in the estuary, 1 per kilometer, and the reservoirs I've shown on the map are the proposed reservoirs for this project, located from the upper reaches of the estuary upstream the river. There's 2 man-made structures in the Kawi River, and these 2 structures constitute a barrier to migration, especially at low tides. These are the 2 man-made structures at Kawi River pictured here during low flow. The weir is quite lower in comparison to the causeway, which is higher up. And if you look closer, you see that it has a fish ladder, which is built to help fish cross to either side of the causeway. So the plan is to put receiver on both sides of the weir and to put other receivers on both sides of the causeway in order to observe any movement in between. There is more than 5 species of mallets present at Kawi Estuary and River at the moment. So in order to prepare for this work, we visited the SAIAB collection facilities and looked at the species of mallets that are present in Kawi River in order to learn to identify them according to morphology. Furthermore, the 20 receivers that are present in the estuary were replaced in preparation for this work. 20 eels and 20 mallets will be collected using rod and line, SAIAB and 5net, and installed with acoustic transmitters. And for the reproduction aspect, bonnets will be collected monthly and preserved in 95% alcohol for histological analysis. We expect the habitat use of both of the species to be related to abiotic factors, and during winter we expect limited home ranges, which is what is shown for other similar species. And during the spawning season, we expect both of them to migrate down to the coastal area and be detected by an array of SAIAB receivers ranging all the way from Western Cape to KwaZulu-Natal. This data will be downloaded every 3 months from the acoustic telemetry receivers, and this will be stored in the laptop for further data analysis. This is the proposed structure of my thesis, and with chapter 2 and 3 being about the spatial temporal movement patterns of both the eels and the mallets, and then chapter 4 linking movement of mallets to the spawning behavior, and then chapter 5 using the use of freshwater, estuarine, and coastal habitats by Catrodroma species in the Kawi River, and then the last chapter being general discussion. I would like to acknowledge the following people and thank you for listening to my talk.
Thank you very much, Dina, for that interesting proposal. Let's see, are there any questions? Remember to put any questions in the chat if you have any questions about Dina's study. Okay, so while we wait for them questions to come in, my question is simply, I don't know, maybe you mentioned it, maybe I might have missed it, but are you sure that some of the species that you are going to tag in your study will actually migrate during the period of your study? Because from my understanding, some of them actually spend some years within the freshwater ecosystems.
Thank you for your question, Tadiwa. No, definitely not. Some of the species can go up to 15 years before they migrate, like eels, for example, and I'm not sure if they will migrate, especially during this period of my study, which will be 1 year. But we hope to just see how they use the estuary, the river, and if they do move, then how would they use the coastal ecosystem during this period.
Thank you for that. We still have time for maybe one last quick question. Okay, last question. In terms of identification, I am sure you're now masters of identification of these species now, or do you go with someone to help you with the identification during your field trips?
Yeah, that's a very interesting one, because at Kawi we have more than 5 species of mallets that we know of, and they all look almost the same, and we rely on experts to help us identify them. But we also went to the SAIAB collection facilities to look at some of the species before we can go to the field in order to get an idea of how they look. Yeah, but we also do take pictures and DNA samples to further confirm our identification.
Okay, brilliant. Thank you very much. I guess we can move on to the next speaker, Enya Manting, and title is Marine Protected Areas for Fish and Fishes, investigating the multi-dimensional effects of KwaZulu-Natal's Marine Protected Area Network on the commercial long line fishing sector. Thank you.
Good morning, everybody, and thank you for coming to my talk on my project proposal for my master's, which is titled Marine Protected Areas for Fish and Fishes, investigating the multidimensional effects of MPAs in KwaZulu-Natal for the commercial line fishing sector. I'm supervised by Dr. Anthony Bernard from SAIAB and co-supervised by Dr. Jess Coburn from the Environmental Science Department at Rhodes, Dr. Bruce Mann from the Oceanographic Research Institute, and Dr. Lauren DeVos from the Save Our Seas Foundation. To cover the contents of this talk, I will first introduce the project, then cover my aims and objectives, my methodology, my budget and time plan, and finally my references before giving a chance for some questions. So the commercial line fishing sector in KwaZulu-Natal is such that total allowable effort is set at 52 vessels and 354 crew. Not all rights are activated each year, and currently only about 30 are active. Most of the rights holders are also boat owners and skippers, and they will generally employ a crew of approximately 5 men. Boats will launch most often in the north from areas near Richards Bay and Tugela Mouth, while in the south they will launch between Port Shepstone and Port Edward often. Fishers generally launch early in the day and will stay out for about 7 hours, and the commercial sector has the highest catch per unit effort of all boat-based line fishing sectors. The commercial line fishers in KZN rely on catching smaller reef residential species when they are most abundant. And this is an interesting response to the crash of certain popular fish stocks in 2000. The National Biodiversity Assessment of 2018, focusing on the marine realm, found that fishing of all types remains the biggest cause of marine biodiversity degradation and the greatest pressure on marine biodiversity, despite mounting pressures from other sources. MPAs with no taxonation are one of the most important tools in fisheries management, and 4 major benefits have been ascribed to MPAs for fisheries. These include fish stock management, improved socio-economic outcomes for local communities, fisheries stability, and the ability to act as ecological offsets. 20 new MPAs were declared in 2020, bringing marine protection up to 5%. In South Africa, in all of our marine protected areas, the recovery of resources is the 3rd most stated socio-economic objective. Interestingly, line fishing intensity is not evenly distributed along our coastline, and the KZN bight, indicated above, is one of the areas that receives some of the highest fishing intensity all around in the country. This is also the location of the 2 largest marine protected areas in the country, and all the other MPAs in KZN are not small either. This leads to a very interesting conflict between the need for recovery of resources and the need to use resources. This conflict is what I wish to explore in my multi-methods research project, which will contain an ecological component and a social component. My project feeds into the larger SAIAB Smart Zone project, which will run for 3 years and aims to collect baseline data for the uThukela Marine Protected Area, which was declared in 2019. The SAIAB Smart Zone project will further feed into the uThukela MPA ecosystem-based adaptation project, which will run for 5 years and aims to improve the resilience of the uThukela MPA to climate change. So to get into more detail about this, I aim to collaboratively build and contribute knowledge on the efficacy of marine protected areas in KwaZulu-Natal for the commercial offshore line fishery by using a multi-methods approach to determine how well the MPAs protect and replenish commercially important fish stocks, and how the MPA network and its current governance affect commercial line fishers. My first objective and my only ecological objective is to build knowledge on the ecological effectiveness of MPAs in KZN for protecting and rebuilding fish stocks that are used by the commercial line fishery. My second objective and my first social objective is to determine how the commercial line fishing community perceives the MPA network in KZN. I will then determine where change may be most impactful for meeting long-term social and ecological MPA sustainability goals, especially in the context of the recent expansion of the MPA network and its associated pressures on fishers. Lastly, I will determine how science communication and participation in research can affect members of the commercial line fishing community with regards to their perceptions of MPAs, MPA research, and the management of marine resources. Moving on to methodology. For the ecological research component, data collection has already been conducted using baited remote underwater stereo video systems, or BRUVs, pictured on the top right, according to standardized protocols, where each BRUV was deployed onto the sea floor for 1 hour with 1 kilogram of crushed sardines used for bait. We used white LED lighting to illuminate fish up to 5 meters away from cameras, as we were sampling at depths between 50 and 200 meters and didn't always have natural light available. We collected 130 samples, comparing various sites inside and outside the Utikela Banks MPA, and comparable data from Isimangaliso, Protea Banks, Alival Shoal, and Pondaland MPAs are already available through SAIAB's MORIP, or Marine Imaging Platform. Data analysis will occur using CGIS event measure stereo software, pictured at the bottom right, to find metrics of population, length, and biomass. I will then use R and R Studio with ANOVAs and t-tests to test for differences between the different locations and management zones. Fish species that I've chosen for the study include the Natal Emperor, black mussel cracker, slinger, Santa, 74, cat-faced rock cod, yellow belly rock cod, red steenbras, and Scotsman. The species that are listed on the left of the picture are those that are currently very important to the commercial fishery, whereas those on the right-hand side are either more sensitive to overfishing or have already been overfished and are therefore interesting to monitor to look for MPA effects with time, especially in older MPAs. For the social components of this research, I will base my approach on the participatory action research approach as far as possible, given the time constraints of a master's. This approach very broadly aims to ensure that participants are involved in the project throughout and are not sidelined after interview data has been collected. Their opinions and thoughts are included in the whole process and contribute meaningfully to the work. Data collection will consist of structured and semi-structured interviews to understand perspectives around MPAs. Snowball sampling will be used and a minimum of 20 participants will be looked for, but ideally all 30 of the commercial right holders will be approached. Interviews will be recorded using voice recorders and video, and the perceptions explored in these interviews will include the ecological effectiveness of MPAs, the social impacts of MPAs, the perceptions of MPA governance, and the perceptions around marine protection. I intend to analyze my data in vivo using thematic analysis. Of course, research communication is a very important part of action research, and I intend to prioritize communication with my participants throughout the research. I will be providing initial feedback to fishers in workshops, where I will also request feedback from them on my findings so that I can ensure that my work and my final report is ground truth to their reality. I will also intend to publish in peer-reviewed journals and would like to credit interested participants as co-authors, as per the tradition of Liebenberg et al. And Jesse Coburn. I will also finally produce a short video sharing the commercial line fishers' perspectives on MPAs or any message that they deem essential that has arisen from the work. Of course, ethics is a very important part of all research, and everyday ethics will be applied throughout the research process, focusing specifically on ensuring the respect and dignity of participants, on transparency and honesty, accountability and responsibility, as well as integrity and academic professionalism on my behalf. Animal ethics was granted in March of this year, and human ethics was approved in the end of October. Okay, so for my budget and time plan, you can see that I have divided my time plan into quarters, where each quarter represents 3 months of the 2 years that I will be spending in my master's. My budget, as you can see, is not small, unfortunately. And the next 4 slides contain my references. Thank you very much for listening to my talk, and I look forward to your questions.
Thank you very much, Enya, for an interesting talk, which I think resonates a lot with what some of our keynote speakers have been saying a lot today. And welcome any questions for Enya in the chat box, or if someone wants to speak live, there's also an option. Okay, while we wait, I just have a small question. I was like, as someone who's just basically done the hard science part of things for the majority of my studies, how hard is it to then actually try and sit down with people and actually get to hear their perceptions? Like, is it harder than actually doing the hard science? It's just a out of curiosity type of question.
I think that's a very good question. Thanks so much. Yeah, it's not harder. I'm in the field at the moment. Starbucks, I'm sorry. But yeah, I've found that all the fishes have been very responsive and very open. They're all very excited to speak to me. They're happy that someone's interested in their perspective on the problem. And they want to share their knowledge. Spoken back so far. I've actually managed to interview 20 fishes already. And so far it seems like all of them are very willing to help. Yeah, there are a few who don't respond, but most of the time it's actually been easy going. And I think that, you know, on the scientific, I think what's difficult is trying to integrate the 2 types of science and trying to do both of them at the same time, because I don't come from social science background, so I've had to learn a lot this year. And I think that's what's really important, just trying to get all these different ways of doing and thinking, trying to get them together in a way that works, you know? That's the hardest thing.
Okay. Thank you very much, Enya. If there are any more questions, I think people will post them into the chat and you can interact. So just keep the conversation going. I think now we'll be breaking for a short comfort break. I'll be back here at 3:50, and Tolana will take over as chair after that. Thank you.
Thank you everyone for coming back to our second session, to our last session of the day. We hope that you enjoyed the following talk. We are still on fish biology and ecology. Our first speaker for the session will be Violet Dupont, and she was speaking on estuarine habitats used by sub-adults and adults of EFS.
Good day, everyone. My name is Violet Dupont, a master's student at Rhodes University. I will be presenting on the estuarine use by sub-adult and adult EFS, one of the chapters from my master's thesis that I recently submitted. The project was supervised by Dr. Taryn Murray and Mr. Matt Parkinson.
Estuaries and coastal regions are widely recognized as highly productive and significant ecosystems that provide a range of habitats for fish and sustain essential ecological connections with other environments. Many marine fish species are estuary dependent, spending a portion of their lives in estuaries, particularly during the juvenile life history stage. On maturation, adults and sometimes sub-adults move out of these estuaries and into the shallow inshore marine environment, where they generally remain for the duration of their adult phase. Despite these typical ontogenetic habitat shifts between juvenile and adult life history phases, adults of some important estuary-dependent fishery species, although few, have been observed frequenting estuaries. This has major management implications for these species, as the estuaries and the associated catchments are among the most modified aquatic environments with habitat loss and habitat destruction posing a threat. Understanding how species use estuarine habitats aids in determining essential habitats and subsequently conservation and management of both the species and the ecosystem. Lychia amnia, commonly known as clearfish or garrick in South Africa, is an estuary-dependent lionfish species of high ecological and recreational importance. The species was decommercialized in 1973, and since then it has only been available for recreational usage. In the early 1990s, a stock assessment for learfish revealed that the spawning stock biomass had been optimally exploited. The daily bag restrictions were reduced from 5 to 2 per person in 2005, and currently the daily bag limit is 2 persons per day, and the minimum size is 70 centimeters. A second stock assessment in 2008 concluded that the stock had collapsed. No new restrictions have been issued as of yet, owing to lack of understanding of how prior regulations influenced the fisheries and lack of movement information on the species. Acoustic telemetry is a popular tool to investigate movements of fishes. It allows simultaneous study of multiple individuals, making it possible to directly study population-level changes of movement patterns. Different movement aspects can be studied using acoustic telemetry, such as habitat use, home range dynamics, estuarine dependency, migrations, and habitat connectivity. Previous acoustic telemetry studies have highlighted high levels of dependency on estuaries by juveniles, but the extent to which larger fish use these environments is largely unknown. The aim of the study was to use passive acoustic telemetry to gain a better understanding of the estuarine use patterns of subadult and adult learfish. The main objectives were to assess the importance of estuaries for estuary and sea-tagged subadult and adult learfish, to determine seasonal patterns in the use of estuaries by migratory and non-migratory subadult and adult, and also to determine the effect of life history stage on estuary visits by estuary and sea-tagged learfish. 69 subadult and adult learfish were tagged along the South African coastline with long-life acoustic transmitters between 2011 and 2020. Prior to statistical analysis, the detections were grouped according to the tagging environment, then life history stages, and movement behavior. In order to address our objectives, to determine the residency to estuaries, we had to use monthly residency indices. For seasonal patterns in estuaries, we use generalized additive models. To look into the effect of life history stage and tagging environment on estuarine visits, we use general linear models with binomial distribution. Temporal changes in estuarine use pattern existed, with subadult and adult recorded in estuaries in January, May, and again post-migration. Some adult fish were recorded overwintering in estuaries instead of undertaking annual longshore migration, evidence of partial migration and skipped spawning. We also found that the use of estuaries was highly dependent on whether the animals were tagged in an estuary or at sea, highlighting the presence of at least 2 contingents, an estuarine contingent and a marine contingent, with the estuarine contingent using estuaries more than the marine contingent. Ontogenetic changes in estuarine use existed, with subadults using estuaries more than the adults. So what does this mean for management? Estuaries are among the most threatened environments, particularly by habitat degradation and overfishing. Since learfish is an important estuary-associated species in South Africa, being used for food security by many subsistence fishers, the conservation and management of these systems is therefore paramount to conserve fishery resources. The long-term dependency on estuary fish returning to estuaries post-migration motivates for increased protection of estuaries, including extending the network of estuarine protected areas in the country, particularly
Western Cape and Eastern Cape estuaries. Thank you for listening to my talk. I'd like to thank my supervisors for academic support, SAIAB for the funding, and all the people that were involved in the project. I truly appreciate you. Thank you.
Thank you, Ria, for your lovely talk. We've got plenty of time for questions. So any questions, you can raise your hand if you want, or you can put them in the chat box. Okay, so a question from Albert, it says, you mentioned existence of marine and estuarine contingents. Can you further unpack this?
Afternoon, everyone. Thanks, Albert, for the question. So what I mean by the marine and estuarine contingent is that we looked into how these clearfish use estuaries, then we further divided them into where they were tagged. So it was either they were tagged in estuaries or they were tagged at sea along the South African coastline. So we analyzed both separately and we found that, and looked into estuarine use for each group. And then we found that the estuarine tagged ones, they use estuaries more than the sea tagged ones. So we could see that according to their behavior, that's how they use these estuaries.
Thank you, Vuyo. Any other questions? I'll ask one. So you said that some of the fish skipped annual migration. Do you have any theories as why they would skip this migration?
Yeah, we do have theories, but we don't have proof. But what we thought would be, in fact, looking into literature, skipped spawning is mainly caused by health issues. So if the fish is not feeling well, then maybe it will skip spawning. Yeah.
Okay. Thank you. Any other questions? Another question from Vuyo, from Albert, sorry. So does this mean there are possibly different stocks?
So there was a study that did like genetic studies on the learfish and found that the South African one is the same stock. So genetically, they are not, they are the same stock. They are not different, but I think it's because of they're different according to their behaviors. So genetically, they're same stock.
Thank you. I think we can move on to the next speaker. Thank you very much, Vuyo.
Thank you, guys.
So our next speaker will be Thembelihle Ndlovu, and she'll be investigating the environmental influences on fish presence and abundance in permanently open warm temperate estuaries.
Hi everyone, my name is Thembelihle Ndlovu. For this presentation, I will take you through my master's research project, which investigate what lives in the Keabums estuary. Estuaries are essential for a large number of fish species due to their shallow and sheltered nature, as well as the food-rich habitats that they offer. However, estuaries are being modified by anthropogenic pressures, as well as changes in the environment linked to climate change, which in turn influences the abundance and diversity of estuary-associated fishes. It is therefore essential that we investigate the changes in fish assemblages in relation to environmental conditions to ensure that estuarine ecosystems are sustainably managed for socio-economic benefits. Moreover, in South Africa, there are almost 300 functional estuaries, and no two estuaries are the same from an abiotic perspective due to their diverse habitats. It is therefore essential that we study what abiotic factors influence the successful recruitment of fishes into these estuaries and to learn how these factors can be managed sustainably. Moreover, we can begin to understand whether an estuary is in a healthy state or whether it's a supporting ground for threatened species or fish species important to local fisheries. So the chosen estuary site for this research project is called the Cape Estuary, which is located in the beautiful town of Gothenburg Bay in the Western Cape province of South Africa. So from this estuary, we have chosen 3 study sites. We have Beto, which is our site 1 located approximately 1 kilometers away from the river mouth. We have Beto 2, which is located approximately 2 kilometers away from the river mouth. And lastly, we have Porkies, which is located at about 3 kilometers away from the river mouth. So the main aim of this research project is to identify and quantify which fish species are present in the Cape estuary and under what environmental conditions they are growing. So the study's specific objectives were to gain an understanding of which fish species were encountered in the estuary and what was their abundance. Secondly, the time spent by the identified fish in the estuary, that is, are they present year-round or seasonally? Thirdly, the location of these fishes in relation to the estuary mouth, that is, are they only present near the estuary mouth or they also occur further up the estuary? And lastly, we want to know the influence of temperature and tidal stage on the presence and abundance of the identified fish. It is important to address this question since the Cape estuary is located adjacent to the sea and is therefore influenced or affected by tides. Also, most fish are regarded as being ectothermic, meaning that they cannot control their own body temperature, but they do so by moving to environments of preferred temperatures. So the remote underwater video system will be used to non-invasively observe and identify the fishes that are present in the Cape estuary. This system consists of a rod with 1 underwater camera mounted on one end, and on the other end is a bait bag of crushed pictures to attract the fish into the camera view. The system is deployed into the estuary for a period of 1 hour, and other environmental parameters such as tidal stage and water temperature are recorded at each study site. So thus far, I've managed to watch 1 year worth of graph footages across all seasons. From these footages, I've managed to identify 30 species that belong to fishing families, and these are represented in big on my screen. We can see that the most common family was the sparrows, which are represented by these red bars. So each of the fish species that I identified, I have further classified them according to their extent to which they depend on estuaries in its life cycle. So the first group are made up of 8 estuarine-dependent species, which consist of estuarine species as well as euryhaline marine fish species. So the estuarine species that we observed were those small fishes, which we call the goby fish, which only breed in estuaries. And then the euryhaline marine species consisted of the Cape Stump-nosed, Learfirst, Cape Mooney, Natal Stump-nosed, white sternbrine, spotted grunter. These fishes usually breed at sea, but their juveniles are dependent on estuaries as nursery grounds. So 6 partially estuarine-dependent species were observed in this estuary, and these were regarded as being partially dependent because they usually breed at sea, and their juveniles may or may not use the estuaries to varying degrees. So the most interesting group to observe in this estuary were the marine species which don't depend on estuaries. And what was more interesting is the fact that they made up majority of the fishes that were observed. They made up 16 of the 30 species that were identified. The top 4 most interesting marine species that were observed included the sienke on your top left corner of your screen. These species usually occur in rocky reefs on the coast of the Western Cape and aren't supposed to be in estuaries. We also have the mass bunker, which occurs in large shoals in the deep sea. However, they are also seen in these shallow estuaries, swimming in large groups. We also have that small yellowfish called the Cape knife jaw, which is supposed to occur in inshore reefs. And that last group of 4 fishes is what we call the greater amberjack or the greater yellowtail fish. These are pelagic fish and occur in the vicinity of deep seaward reefs. So the question is that what are they doing in the estuary? So this slide answers the question of where these fish species were located in the estuary. We can see that all 4 estuarine dependent categories were observed in all of our 3 study sites. So from our graph footage, we'd estimate the max N for each of the observed or identified fish species, and the max N is equivalent to the maximum number of individuals belonging to a particular fish species. So this is an interesting figure which shows the changes in max N of the most common fish species, and the fish species with the highest max N of 90 individuals was the mass bunker, which is scientifically known as Tricharis tricharis. As mentioned before, these usually occur or swim in large shoals. So seasonal and spatial variations in species richness was observed in this estuary, with the winter season having the lowest number of species and the species richness reaching a peak during the autumn season. It was also observed that porkies always had the highest number of species across all seasons, and Beto 2 always having the lowest number of species across all seasons. So the species richness tends to decrease as you move from porkies further up the estuary across all seasons. So we already know what was found and where it was located. So this slide shows the season in which all the identified fish species were observed, and both estuarine and marine fish species were observed across all 4 seasons, with the marine species consisting or making up majority of the fishes across all seasons. So having discovered which fish species were present in the estuary, where it was located and when it was located, the next question to address is, can this be correlated to temperature? So from our temperature recordings, the figure below shows that warmer temperatures were experienced or occurred during the summer season, with cooler temperatures occurring in the winter season. It was also found that Turkey, which lies next to the estuary mouth, was always warmer compared to the other 2 study sites, which lie further up the estuary river mouth. So the presence of threatened species, according to the IUCN Red List of threatened species, such as the vulnerable white stump-nose, vulnerable elffish, and the endangered white stiambra, as well as the critically endangered common stingray, suggests to us that this estuary is an important area for conservation. The seasonal variations in fish assemblages could be a result of the migration of species out the system as they grow and the arrival of new ones. For instance, during the winter season, most but not all of the elf and the leaffish fish species will migrate northward to the KZN coastline to spawn. An influx of these juvenile fishes would then migrate back into the estuary during the summer and spring season. Spatial variations could be attributed to temperature and salinity changes. So the highest number of marine species observed year-round at Porkies, which lies next to the permanently open estuary mouth, may have reflected the relatively high salinity conditions that exist in this study site, as well as the warmer temperatures that exist in this location. In contrast, the low species richness at Beto 2, which lies further up the estuary, could be attributed to the low salinity condition, as well as the cooler temperatures that exist in this study site. That brings us to the end of my presentation. Thank you everybody for listening. A special thanks to NRF SAIAB for funding this research project. And thank you to my supervisors, and yes to discovering what lives in the caverns.
Thank you very much, Temilisha, for your talk. We already have a question. It says, hi, Temilisha, do you think baiting your cameras is skewing the dominance of your marine predators And were your sites in the channel or in the littoral margins of the estuary?
Hi, Nicola. That's a very good question. And that's something I have to consider. Not quite sure how to answer this question because I'm not quite certain.
Okay. Thank you. Perhaps you'll go and find out, or further studies will help you answer that question. We have another question by Anthony, who says, very nice talk. Are there plans to use stereo browse to try and get some size data for a species? It would be interesting to know which size classes of the typical marine species are used in the estuary.
Okay, thanks for that question, Anthony. I have been watching the graphs for the entire, and since I'm only focusing on identifying the species that occur in this estuary, it would be really interesting to also know what are the life stages of the fishes that I've identified. So that's definitely something that I might consider diving into if I further my studies. But for this particular project, I won't be looking at the life stages of the fishes, just only looking at presence and abundance.
Thank you, Temelise. There are other questions for you in the chat box, which you can attend to. Would like to also remind people that the link for voting for the best presentations is now open again, and you can go and vote. So our next presenter will be Bilon Tlenyawo, and she'll be talking on using environmental DNA and stereo browse to assess the influence of depth on reef fish community structures.
Greetings to all. My name is Bilon Tlenyawo, and I will be talking about the evaluation of the application of environmental DNA for the assessment of reef fish community structure. Marine ecosystems and the substantial biodiversity they support under increasing threats from indirect and direct human disturbances. With the increase in these threats, there's a recognition that scientific sampling needs to be both non-invasive and non-destructive, eliminating the potential damage to marine communities and habitats while providing a realistic insight into the diversity and ecosystem relationships. There is a growing interest in the use of environmental DNA as a tool to conduct fish surveys in the aquatic environment. Specifically, environmental DNA is rapidly gaining interest as a tool to conduct fish surveys, assess distribution patterns, and conduct spatial ecological research in the marine environment. So what is environmental DNA, or eDNA for short? Environmental DNA is defined as the DNA that can be extracted from the environmental samples, such as soil, sediment, and water, without first isolating any target species. With that being said, environmental DNA is considered as a non-invasive sampling tool that provides data from the diverse mixture of genomic DNA shared into the environment by numerous species. Furthermore, in comparison to traditional monitoring techniques, such as stereo browse, that are well calibrated and well established, sampling environmental DNA is straightforward, with typically 1 or 2 liters of water collected per sample. Even though environmental DNA has emerged as a potentially powerful tool, it is still a relatively new tool, and the method would benefit from more field implementation that evaluates its effectiveness and practicality as a marine biodiversity monitoring tool, especially in the South African context. The aim of this study is to then determine if environmental DNA can be used to analyze spatial and temporal patterns in reef fish biodiversity and community structure through field experiments and comparisons with baited remote underwater stereo video systems. The main objectives of the study are to develop and standardize a protocol for assessing marine fish assemblage structure in offshore reefs using environmental DNA, evaluate if environmental DNA can be used as a tool for the assessment of reef fish community structure inside and outside marine protected areas, assess the vertical migration of reef fish assemblages using environmental DNA and stereographs, and lastly, use environmental DNA and stereographs to assess the influence of depth on reef fish community structure. And this is the objective I will give preliminary results of. Sampling for the study was conducted in 3 locations, but today I'm going to present data from Bird Island Marine Protected Area and the Tuticoa National Park Marine Protected Area. 16 replicates were collected from each location, consisting of 8 replicates in the shallow reef and 8 replicates in the deep reef site. In each site, water was collected at the surface, then at the bottom for environmental DNA surveys, and then at the same site, the stereograph was left to record for an hour on the seafloor. All DNA extractions were carried out using the eDNA tissue DNA kit with few modifications to the original manufacturer's protocol. PCR was conducted using the index primers targeting 240 base pair region with the 16X fragment. And then samples were sent for sequencing. The dataset curation and taxonomic assignment of sequence results were conducted on mother and BLASTM. Finally, all stereograph samples were analyzed using the events measure software. For statistical analysis, generalized linear models, or GLMs, were performed to test the effects of method and depth on diversity indices, and the community analysis was conducted using Primer version 7. Just to give a brief overview of the preliminary findings around GLMs for species richness and relative abundance, the preliminary results indicated that there's no significant difference in richness in stereographs and environmental DNA bottom. However, stereographs and sampling water at the bottom detected more fish species than the water samples collected at the surface. There was also no effect of depth in all the methods. The interaction effects of method and depth showed opposite patterns for environmental DNA bottom and stereographs, and interestingly showed a similar pattern for environmental DNA surface and stereograph. Basically, a similar trend was observed for relative abundance. It is important to emphasize that the results are bound to change once I figure out how to best display the results after the analysis is complete and the abundance from the stereographs and eDNA surveys are standardized and made comparable. Also, the species richness output is not a fair comparison as the reference library is still in progress. However, looking at the trend or the pattern in the trends is important as it provides a useful way of indicating if the methods are showing the same information, and from these preliminary results, they are not. Results from the Permanov analysis of present FSEP data indicated a significant effect of depth, method, and the different locations, as well as the interaction between depth and method. Additionally, the CAP analysis illustrated a clear separation of samples according to sampling method. A simple analysis was conducted to show the common or distinct occurring fish species detected by the 3 methods. For the shallow sites, the simple analysis showed a common set of species for environmental DNA surface and environmental DNA bottom, such as Diplodocus capensis and Chilodactylus species. Species like Bopsodea inanata was common in all 3 methods. Proderma africanum was distinct to stereographs, emphasizing the importance of the reference library, as the shafts have not been barcoded, but will be soon as we've obtained the fin clips. For the deep sites, the simple analysis showed a common set of fish species for environmental DNA bottom and stereographs, such as Chrysoplevis lenticeps, Panga, and Copenta. The squalor species was distinct to environmental DNA bottom and was not detected by stereographs or environmental DNA surface. The most contributing species for environmental DNA surface was the Chilodactylus species, which was also detected by environmental DNA bottom, but not stereographs. So in conclusion, globally, environmental DNA is getting support as a sampling tool due to its ability to effectively collect biodiversity information over large spatial scales. This research specifically aims to address questions related to field and laboratory protocols and determine its ability to collect ecologically meaningful data to answer applied questions over finer spatial and temporal scales. Our preliminary results showed that stereographs and environmental DNA illustrated different diversity patterns and that there was a clear separation effect of method and depth on the fish assemblages. However, an accurate, comprehensive reference library is important to maximize the information gained from environmental DNA to allow for unbiased comparisons. Thank you so much for lending me your ear and listening to my talk. I would like to thank the funders for making this research possible. Thanks.
Thank you so much, Mpilo, for that interesting talk. We have a question from Nobuhle that says, that was such a nice talk, nice presentation, Mpilo. I just have two questions for you. One, I might have missed this, but how did you sample your eDNA samples? And two, why compare eDNA with stereographs as there are many well-calibrated sampling techniques, as you mentioned?
Hi, everyone. Sorry for the sound. And normally you didn't miss it. I didn't mention it. But sampling environmental DNA is very straightforward. For me, at each site, I collected 4 liters of water at the desired depth using a Niskin bottle. And at the surface, I scooped 4 liters of water using the Nalgene bottles. And this was done, this was done before the deployment of the stereograph at the same site. So for the second question, so as I said, stereographs have been used globally, and not to say they are without biases, as Thembelihle mentioned in her presentation, they can be costly, and also processing the video samples can also be time consuming. However, many studies have shown that stereographs, to be highly effective at surveying fish communities across depth range, broad depth range, and also different habitats. Most importantly, they cause little to no damage in marine habitats, meaning that they are minimally invasive and can be used to survey and document the fish populations in fragile habitats and also in marine areas.
Thank you. Just one more question. It says, thank you for your presentation. I'm curious, what factors could affect your eDNA sample, such as release rate or eDNA persistent? How does dirty water affect your analysis?
So thanks, Ma. So the release rate of, I haven't looked at it specifically for my studies, but the release rate of eDNA of fish into the aquatic environment depends on many factors. For example, a study was looking at the effect of the release rate of fish according to their life stages, and it showed that adults tended to have higher release rates than juveniles in many fish species. Also, as well as the fish persistence, also depend on many factors in the marine environment. For example, depending on the environment or the site being studied, it has been shown or found that environmental DNA molecules degrade faster in the shallow inshore sites compared to offshore, due to many reasons, such as freshwater inputs if you're near a river, or pH levels. So yeah, I don't know if I answered that one.
Thank you, Mpilo. Our next speaker is Samantha O'Case, and she'll be talking about connectivity patterns of slinger between marine protected areas and surrounding areas along the east coast of South Africa.
Good day, I'm Samantha Orkes, and I'll be taking you through a chapter of my thesis, which is entitled Connectivity of the Slinger, Chrysoprepus puniceus between marine protected areas and surrounding areas along the east coast of South Africa. Marine protected areas, or MPAs, are defined as areas of coastline or ocean, which are protected for the benefit of people and nature. Globally, marine protected areas are implemented for the conservation of biodiversity and fisheries management. Initially, coastal MPAs in South Africa were implemented to protect intertidal resources for purposes of biodiversity and conservation, and only later the protection of offshore resources was considered, and the potential role of MPAs in fisheries was taken into account. As of 2019, there is 42 MPAs found around the South African mainland and islands, protecting 5% of the exclusive economic zone. The benefits of these MPAs are seen in increases in abundances and sizes of target species. 5 MPAs exist along the east coast of South Africa, from the northern KwaZulu-Natal coast to the northern Eastern Cape. These include Isimangaliso, Lutikela, Aliwalso, Protea Banks, and Ponderlane. It has been established that these East Coast MPAs conserve the inhabitants and associated habitats. However, there is limited evidence on whether they are connected, form a network, and offer benefits beyond their boundaries, replenishing the surrounding areas and promoting persistence in marine ecosystems. For MPAs to realize their full potential, they need to be either self-sustaining or connected to individual MPAs through either larval dispersal or adult spillover. Connectivity, which refers to the demographic linking of individuals between local populations, occurs through larval dispersal and adult spillover. Adult spillover has been demonstrated in several studies. However, measuring larval dispersal still faces many challenges, which includes difficulties in tracking small larvae and the large spatial and temporal variation in larval production and survivorship. Connectivity is also a key ecological factor to consider when designing protected areas, as it promotes persistence of metapopulations, the recovery from disturbance, as well as the productivity in marine ecosystems. In the past, connectivity has rarely been taken in consideration when designing MPAs, as the focus were more on conservation. To test for connectivity among East Coast MPAs and surrounding areas, we used the Seabrim Chrysoprepus puniceus, commonly known as the slinger. The slinger is endemic to the southern Mozambique and the east coast of South Africa, with core distribution between Ponta Savona in Mozambique and Coffee Bay in the Eastern Cape. This species is the major component of the commercial line fishery and recreational ski boat fishery of KwaZulu-Natal and the southern Mozambique. The slinger is mostly resident, however, occasional long-distance northward movements of adults have been revealed in tagging studies. The spawning of slinger is restricted to the northern reefs of KwaZulu-Natal and the southern Mozambique, where it occurs in shoals, producing small pelagic eggs containing an oil droplet, which provides the eggs with extra buoyancy and increased dispersal potential. Initial assessments indicated that the Agulhas current is responsible for southward distribution of recruits on the east coast. However, the typical speed of the Agulhas current is between 1 to 2 meters per second, and juveniles would be transported too far south for successful recruitment. Subsequent research indicated that processes at the forward edge of the Agulhas current is responsible for retaining lionfish larvae on the south or southward dispersal. The strategy was also suggested for the slinger in a study by Duncan et al. 2015, where southward dispersal were assisted in soar of the Agulhas current. The importance of slinger as a fishery species and its life history characteristics makes the species suitable to examine genetic connectivity between East Coast MPAs and the surrounding areas. Therefore, the aim of the study was to examine genetic structure and gene flow in the slinger between MPAs and unprotected areas along the east coast of South Africa. To examine genetic structure and gene flow between MPAs in the surrounding areas, fun clips of about 100 adult slinger were sampled at 5 sites. These included 3 MPAs, Isimangaliso, Itukela, and Alewelso, and 2 unprotected areas, Richards Bay and Port Edward. The study also forms part of the funded capital project, which examined connectivity between East Coast MPAs. In the lab, DNA from fun clips were extracted using the sorting out protocol. Restriction site-associated DNA sequencing, or RAD sequencing, was used to generate single nucleotide polymorphisms. For this particular study, we used quad-RAD sequencing. RAD sequencing can generate thousands to 10,000s of random fragments across the genome, covering a great depth of coverage, which makes this method robust and efficient for genotyping a large number of loci with high accuracy. Diversity estimates were calculated in the populations module and the stacks pipeline. Overall, there was no clear patterns in genetic diversity among marine protected areas and unprotected areas. Population differentiation among sampling sites was investigated using pairwise FST comparisons. A principal component analysis was performed to examine patterns of genetic differentiation or similarity between sites. Pairwise FST values among sites was low and non-significant. The PZA results revealed no clustering based on geographic origin of samples, implying high connectivity among MPAs and unprotected areas. To infer population structure among sites, we used 2 methods, structure and fine RAD structure. Structure analysis revealed most likely number of genetic clusters to be 2. However, all individuals showed similar degrees of admixture of the genetic clusters, indicating a lack of genetic structure and implying genetic connectivity among MPAs and unprotected areas. Fine RAD structure groups individuals with high level of shared co-ancestry. Colors indicate scale of relatedness between individuals, with yellow indicating low relatedness and blue or black indicating high relatedness. In this heat map, no geographical structure among MPAs and unprotected areas were found. There was no pattern of shared co-ancestry based on geographic origin of each sample. Bayes was used to examine gene flow among sites. Bayes indicated multi-directional migration rates among MPAs and unprotected areas. The highest migration rates was recorded from Richards Bay to all other sites, with low migration rates in the opposite direction, identifying Richards Bay as a key source population. There was also moderate migration rates recorded from Singwazi to Scottburgh and from Port Edward to Singwazi. The net migration rates were predominantly in a southward direction and likely influenced by the southward flowing Agulhas current. Overall, the main findings of this thesis indicated a lack of genetic structure among slinger populations along the KwaZulu-Natal coast, implying high levels of gene flow among MPAs and adjacent unprotected areas. Furthermore, multidirectional migration rates were observed between sites along the coast, with the highest migration rates recorded southwards, which is probably influenced by the prevailing southward flowing Agulhas current. As such, MPAs along the KwaZulu-Natal coast are connected and function as a network, and more likely replenishing adjacent unprotected areas through larval dispersal and adult spillover. This study reinforces the significance of necessity for effective management of MPAs along the East Coast, which serves as spawning and nursery grounds for this important fisheries species. I would like to thank the following people who made this study possible, and thank you for taking the time to listen to my talk.
Thank you, Sam, for that wonderful talk. We've got time for just one question. Okay, thank you. So I'll ask you a question. So Sam, do you think that since the marine protected areas, then since there are so many marine protected areas, do you think it's worth having them? Like you had five marine protected areas. If one of them is feeding the whole other regions, do we necessarily need all of them?
So for why I chose those sites or whatever, it's where my fish occurs. And then, yeah, so how can I say, so yeah, that's why we chose, because I mean, and those impacts were all like implemented for several reasons with overexploited fishes that occurs in that sort of areas. So that's why they came and sort of, so we just trying to see whether they are connected and they actually doing way more than just protecting that area, maybe replenishing the surrounding fished areas. Yeah.
Thank you, Sam. You've got a couple of comments in the chat box, so you can attend to them. Thank you. So our next speaker is Angus van Beek, and he will be speaking about understanding the and complementary of the disparate fish visual sensors techniques and optimizing data value for stakeholders.
Good afternoon, everybody. My name is Angus van Beek, and today I'll be presenting a brief background and some preliminary results from my PhD research titled Understanding the Interoperability and Complementarity of Disparate Fish Visual Sensors Techniques and Optimizing Data Value for Stakeholders. So just a brief background for the project. When we talk about fish visual sensors techniques, we generally discuss a handful of the primary techniques. So these include baited remote underwater video systems, diver operated video or underwater visual sensors, and remotely operated vehicles or ROVs. So historically, although there have been a number of papers comparing the different techniques, the majority of marine research has been conducted using these methods somewhat independently. However, more recently, there's been a focus on trying to better understand the complementarity of the different techniques and how to best use them in conjunction to better understand our marine environments. So what then do we mean when we talk about interoperability and complementarity? Well, we can look at these three different techniques and their monitoring strengths. If we're attempting to monitor a reef with varying depth profiles, for example, it may be beneficial to use probes to quantify elasmobranchs in fishery target species, use diver operated video to discover high numbers of those small benthic species, and ROVs to survey deeper reef gradients in deeper waters. The complement of data should provide a more robust understanding of the studied ecosystem. So what we're aiming to achieve in this project is to gain a much better understanding of the global community who uses underwater visual sensors methods and how their combined data sets may be put to use. And this will allow us to develop our own framework from the ground up with collectors of visual sensors data and other interested stakeholders. And ultimately, we then want to figure out what is the best way to deliver this data as long-term data sets to global networks, such as MBON or OBIS. What I would like to present today is twofold. Firstly, objective one, which is to understand the Visual Sensors Global Research Network. And the second is to highlight one of the many fantastic open source tools available to our students. We talk about understanding the Global Research Network in a somewhat simplified manner, looking to understand who is involved, where is the research being conducted, and what exactly is being studied. And ultimately, we would like to understand how do these different methods coexist within a research landscape. To gain an understanding of the research landscape, we decided to make use of Cortex, an online script manager to conduct an epistemic analysis. The first step to carry out the analysis was to curate a large corpus, or basically a large collection of written text. So we did this by doing an online search for pre-selected terms and downloading the bibliographical data. This is a similar process to what you'll do for a systematic review, but we were also interested in collecting more texts like abstract text and research affiliations. We collected over 1,400 data points, which we used as the corpus and fed this into core text. Without going into too much detail, Cortex makes use of natural language processing to extract the most salient multi-terms from large bodies of text. So, for example, by using parts of speech tagging, chunking, normalizing, and statistics to discern between nouns, verbs, and non-important phrases. We would only be interested in, say, joint nouns, reef fish, for example, especially if mentioned many times in the text, versus, say, the fish swims fast, which includes nouns, verbs, and adverbs, and may only be mentioned a handful of times. This we're not interested in and would be excluded from the analysis. Once we uploaded our corpus to the Cortex manager, we were able to take on our first question using a script to run geocoding. So, where and who is conducting underwater visual census research? We used institutional research affiliations for each publication, and we geocoded these. So, for example, the Marine Technology Center in Japan, from this inconsistent body of text, was able to be geocoded by core text and determined to be in the country Japan. So this is particularly powerful because with the 1,400 plus or minus publications we had, there were roughly 4,000 unstandardized locations, and these could be geocoded within a few minutes. So the count of these affiliations could then be displayed on this interactive map along with the visual census techniques as a variable. So when who is conducting underwater visual research? Well, it's predominantly conducted by developed nations, including Australia, primarily involved in browser research, Europe, and the United States of America, who conducts a lot of ROV surveys. We then went back to the Cortex manager, and there is salient multi-terms that we extracted from our corpus abstract text. And we made use of the network mapping script to define and graphically display the underwater visual research landscape.
This is the network analysis for visual census research based on those terms we extracted from the abstract text of our corpus. What you're seeing here are a number of nodes connected by edges. In a nutshell, the size of the circular nodes displays a measure of importance, whereas the distances between the nodes and the edge lines displays a level of connectedness. So when the nodes cluster together like this and the edge lines shorten, you begin to witness what are called knowledge clusters within a research landscape. So for visual census research, there seems to be 8 distinct but connected knowledge clusters. So these are natural reefs and artificial structures, such as artificial reef or underwater gas pipelines. There is research looking into submarine canyons and demersal fish assemblages, so some deep water research. There's a knowledge cluster upon shark research. There's a strong proponent of reef research within the community. There is a somewhat independent method research knowledge cluster that we're observing in the top left. There is strong representation of marine reserves and no-take areas within the research landscape. Then there's a somewhat independent and isolated seagrass habitat research going on. And the same can be said for herbivorous fish species and their relatedness to reef in the bottom right. One of the more powerful things we found by using an interactive manager such as Cortex is the ability to investigate your data further visually. So within the network, if we focus on shark research, for example, we can see which other areas of research sharks are related to and its level of connectedness. So marine protected areas, habitat associations, management plans, fish sites, conservation efforts, and extinction risk. And this discourse is completely different to that of reef fish and the research being conducted within its knowledge cluster, which has a focus on community abundance and diversity studies, for example.
Once we had built this network, what we were able to do was add the different visual census techniques as a variable to the existing framework. So you can see the top contributing underwater visual sensor technique for each knowledge cluster. So RUBs have played an important role in marine protected area no-take research and in elasmobank and shark research, as well as assessing deeper water habitat. Whereas ROVs have played a critical role in artificial reef research and surveying deep water artificial structures. And underwater visual sensors has played a leading role in the reef research to date. So in conclusion, what we are saying is that the underwater visual sensors research landscape is incredibly interconnected, and that the different techniques have each played a role in forging unique knowledge clusters. So our next objective is to develop an online questionnaire and a multilingual knowledge base to better understand the individuals within the network and how this community compares to the research landscape we derived from their published work. Great, thank you so much for attending my talk. And yeah, I look forward to any questions anybody might have about Cortex or about our project. Thank you again.
Thank you, Angus, for the talk, for this wonderful talk. Any questions for Angus? Okay, I'll ask a question. So you saw that they were using different visual techniques for different areas. Are you planning on, what is your next step? Are you planning on using two visual techniques at the same side to see if there's comparability or which one is better?
Can you hear me?
Yes.
Oh, great. Yeah, so I'll be using existing data. So looking at individual case studies. So for example, if you look at a single area, like a marine protected area in Namibia, and I'll be looking at, say, available data from a whole bunch of different techniques, say on the west coast of Africa, and then I'll be drawing comparisons of those data. So when you talk about interoperability, it'll be sort of how can we use these different techniques that might have been collected at different times, for example, to hone in on, say, a single metric of abundance. Could be an example. So I won't be trying to understand if each of them are better than one another. It'll be more trying to see if I can use multiple methods to answer, say, a single, or try and derive a single biological indicator.
Perfect. Thank you, Angus. Our next speaker will be Bantoni Zigo, and he will be talking about movement behavior and physiology of the striped mallets.
Hello, everyone. My name is Bantoni, and I'm going to be sharing with you part of my research project, which is on the movement behavior and the physiology of the striped mallet in the Kawi estuary. And my supervisors are Dr. Tari Mari and Professor Emma Child. So to start off, a little bit of background, estuaries, which we all know that they are important habitats for fish because they are highly productive, they are food-rich areas. And unfortunately, these are threatened due to various activities from habitat loss and degradation and overexploitation resulting from activities such as overfishing. And because of that, there's been loss of habitats and also the biodiversity that they contain. And because of these, there is need for a sustainable management of the estuary resources, that is both the habitats and also the fish. Now, in terms of the fish fauna that are found in the estuaries, they need to be information that regarding the biology of the animals, how they interact with other animals, and also the habitat use patterns by these fishes. And also we need to understand the movements, whether they be resident species, they be migratory species, and all of that information is important for us to be able to devise management strategies. And for this research, we'll be focusing on the movements component, which has been studied mostly using acoustic telemetry. This is the most popular method that has been used worldwide to study the movement of aquatic organisms. So the study species, which is the striped mullet, this is the species that is partially dependent on the estuarine environment. So it stays in the estuary and sometimes it goes over to the sea. And this is endemic to Southern Africa. So its distribution ranges from the southern part of Angola, going all the way to the northern part of Mozambique. And a lot of mullet species, including the striped mullet, they have vast ecological importance because of the way they are positioned in the food chain. So they offer food for predatory species, and they are also found in very large numbers in the estuaries, and they also have ecological importance in terms of providing food for people, and also they're targeted as bait for recreational fisheries. And in South Africa, these have been regulated to 50 fish per person per day. And what we found in literature is that this number hasn't been really justified, just because we don't know more information about these species. So, for example, we don't know the movement, their reproduction, and also their physiology. So that brings us to the aim of this project. So for this project, we're going to understand the movement of these species by comparing the movement of juvenile and sub-adult striped mullet. And we're also going to look at the factors, the environmental factors that affect the movement of this species, which is the environmental variables such as temperature, salinity, and river flow. And we're also going to look at the upper thermal tolerance range of the striped mullet. This is because of the increasing global warming. So we want to understand how these species are going to perform under increased temperature scenarios. So the study area for this project is the Kawi Estuary, which is located in the Port Alfred, Eastern Cape, South Africa. So the Kawi Estuary is a 21 kilometer estuary from the mouth going upstream, and with a width of just less than 150 meters. And the mouth area of this estuary has been highly modified into a marina. So there's a lot of man-made structures around the mouth of the estuary. And the method that we'll be using to monitor these movements, acoustic telemetry. Already we have placed, I'm going to explain later on the acoustic receivers, but already we've placed 20 receivers in the estuary from the mouth going upstream, and this covering about 20 kilometers of the estuary. And these are spaced about 1 kilometer from each other. So acoustic telemetry as a method, we use acoustic receivers. These are the ones that we placed in the water. so that they detect movements of fish that have acoustic tags in them. So once you capture the fish in this study, we are using SAIAB nets. You will set a tag into the fish, perform a little surgery, and then let them recover and roam around. And after that, there's the monitoring process, which is as the fish are moving, if they are within the range of the receivers, their movements are logged onto the receivers, and then you can come back and download the data for that species. And in the field, we'll also be collecting fins and also otoliths. And if we manage, we're also looking forward to collect gonads also in the for the adults, just to understand their reproduction. So for the physiology component, what you're looking for is how the species is able to tolerate increased temperature. So we're targeting 30 species, juvenile fish, which will be placed in batches with different acclimation temperatures. And then so this just the picture just shows the design of not the exact equipment that we're going to use, but we're going to increase the temperature as the fish will be in those water baths. And then as you increase the temperature, you check how many how are they responding and how many will be left after a certain temperature to determine the critical, the more maximum, which is the CT max. So for our results, we expect to see that there will be a high residence of juveniles and sub-adults within the estuary. And the sub-adults, the juveniles are more likely to be present in the estuary than the sub-adults. And also we're expecting to see temperature and salinity to drive this movement. As temperature and salinity, they are also one of the important drivers for other species. And we're also hoping that to see that the reproduction is going to drive certain movements by comparing the movements of adults if we manage to catch adults. So this is structure. We have a general introduction, and we have materials and methods, reproduction involved. We also test the thermal tolerance range of juveniles, and we have a chapter on the movement behavior of the street manners. So we compare the juveniles and the sub-adults. Then we discuss everything in a separate chapter. So I would like to thank my supervisors for this opportunity for this project, and I have SAIAB for providing funding, and Nick Parkinson, who is also involved in the ATIP group for tagging these species.
Thank you, Anthony, for the wonderful talk. We have time for just one question, quick one. You can raise your hand. How long have you been monitoring the fishes that you are monitoring their movements? Or will it be newly tagged fishes?
Oh, okay. Thank you, Tolwan. Yeah, actually, this I forgot to mention that this is a research proposal. So the project just started this year, and so far we've only managed to take one fish. So we're going to be monitoring for the next year. So it's going to be 1 year monitoring process.
Okay, thank you. Thank you, Bantoni. You can, if you want to ask Bantoni more questions, you can give them in the chat. Our last speaker for the day is Thamsanqa Wanda, and he will be talking about geomorphological variability across the Tugela MPA region.
Greetings to all. My name is Thamsanqa Wanda, Durban PhD student with SAIAB. registered at the Nelson Mandela University. Today, we'll look at the presence of reef complexes and its geomorphological variation across the Otsugela MPA region as a basis for numerous niches supporting shelf biodiversity. With the advanced high-resolution imagery of the seafloor, its structural characteristic features that were previously undocumented or not observed when using the old data acquisition techniques such as single beam bathymetry can now be observed, to be documented, and classified accordingly with the new multi-beam bathymetry data. Globally, the seafloor mapping developments are in support of marine spatial planning and management of the marine space, to provide better understanding of the spatial and temporal habitat use, which is essential for a successful conservation process and understanding of the shelf ecosystem's functioning. This provides the ease of better planning and decision-making when the current environmental and societal needs are being addressed, whilst also envisioning a sustainable future. The area of interest is on the east coast of South Africa, offshore central KwaZulu-Natal. The left-hand side image shows the 14 selected sites of survey indicated by the black blocks, which were selected based on being known recreational fishing areas most likely to be reefs, and because of their relative location to the MPA zones, with some of these sites being immediately adjacent to the Otsugela MPA. Currently, 12 of these sites have been surveyed at an average coverage of 15 square kilometers per site. These sites were selected to cover the different varying levels of restrictions within the MPA zones, with the interest of acquiring the seafloor geomorphology, looking into the way in which it interacts with the hydrodynamics and seeing how this forms the foundation for the existing marine ecosystems and habitats within the appropriate scale. The GMAP, which is the geophysics mapping platform vessel, was used during the data collection phase, as can be seen on the top right-hand side image with the fitted multi-beam echosounder equipment. And on the bottom right-hand side image, this shows the behind the scenes before surveys are done, where prior to heading out to sea, one needs to plan out the survey lines within the destined block, and then more other technical planning, such as selecting a good mapping weather window needs to take place, which is essential, and this gives guidance on how each specific site and when it can be tackled. The purpose of this study is to discuss the value and importance of applying geophysical mapping tools in marine spatial initiatives. This will be done by looking into the preliminary results from the Otsugela MPA and its adjacent hydroacoustic survey sites, where I have analyzed the geomorphological variations such as slope or rugosity and the preservation of the reef complexes within the study area to decipher their possible likely contribution to the shelf biodiversity and its ecosystem functioning. Despite considerable progress in terms of marine spatial planning, South Africa still lags behind global leaders in implementing large regional hydroacoustic surveys to cover the seafloor area within the exclusive economic zone. The lack of hydroacoustic data is perhaps due to relative lack of funds, poor communication and understanding with regards to multi-beam echosounders. Therefore, South Africa's seafloor geomorphology remains underrepresented in literature and is only classified at low resolution. As can be observed in the images, this technique uses sound to collect thousands to millions of georeference depth soundings, which are processed to create a 3D digital terrain model of the surveyed area. The multi-beam echosounder captures high spatial resolution data of the seafloor geomorphology used to characterize the marine habitats in terms of their georeference location, spatial extent, and geomorphological characteristics. The multi-beam echosounder collects the depth soundings beneath the vessel as it sails along the ocean, along the planned survey lines. And after processing this collected data through software such as HyPack and Surfer, one then can obtain outputs as the visible examples in the bottom right-hand side image. This is all made possible by the system or system components working in sync with one another. Looking at the environments and regional settings of the shelf, the East Coast continental shelf of South Africa is wave-dominated, with the Agulhas current being the major influence behind the sediment migration along the shelf. Cementation process along the shelf can be very rapid at times, outpacing the rate of sea level rise. Well-cemented deposits greatly improve the preservation potential of the paleo shorelines as sea level increases. The preserved deposits contribute to the prominent consolidated hydrologies on the continental shelf and are expressed as the linear coast parallel submerged beach rocks and eolonite complexes of the late Pleistocene age. These outcrops represent the majority of the present-day reefs along the mid-shelf of the East Coast. These lithologies crop out of the unconsolidated Holocene sediments. Mobile sediments cover the low relief reef or outcrops in places. Let's look at some of the Otuela MPA hydroacoustic survey sites preliminary results. I will be using 3 sites to show the structural variance in the seafloor characteristics, using one example from the south, from the central, and from the northern regions of my study area. One can already see the structural differences in appearances. The reef and paleo shorelines have a degree of variation where in the central zone, it appears to be patchy or mottled with indistinct paleo shorelines. This is different to the northern, which is offshore Richards Bay, and southern, which is offshore Durban or Mdloti, where it is very distinct paleo shorelines and has a rugged reef relief. Northern sides tends to be narrow in across short dimensions, whereas the southern sides host wider regions of outcrop. The rugged varying geomorphology could prove suitable for various compatible biological natures where the high relief reef could provide stable habitats for long-term inhabitants, whereas the low relief reef can provide habitats for short-term inhabitants, as it is more vulnerable to the burial by migrating shelf sediments. The variation in surface geomorphology or morphology is even more visible when the surfaces are compared on cross-sectional profiles. In this southern site, we notice a slope angle of about 10 degrees with a greater change in depth in the distal offshore side of the site. And in our bathymetry, we note the evident paleo shorelines and sediment-starved parts of the survey sites having exposed reef. In the central site's bathymetry, we noticed patchy seafloor outcrop with no visible distinct paleo shorelines. It is likely that these areas were either less well cemented or faced greater exposure to wave revealment that the northern and southern sites didn't experience. In the cross-sectional profile, a slope angle that is less than 2 degrees is observed, suggesting a relatively flat site, which has occasionally outcropping reef at low relief of about 2 meters above the adjacent sediments. In this northern site bathymetry, we noticed that the sediment-rich parts of the site even have bed forms formed on it. And also we noticed that there are prominent paleo shorelines, which are also visible in the cross-sectional profiles as these huge depth drop-off zones with sudden depth changes values of about 10 meters. This 10-meter drop could be a result of exposure to the outer shelf to the Agulhas current. The slope angles that is seen in this site is 5 degrees, which differs from the other 2 previous site examples. In conclusion, with multiple sounders, world representation of the seafloor is achieved. Our results prove the effectiveness and potential of multiple sounders in the analysis of reefs and its adjacent local scale environmental settings, highlighting the importance of how mapping these reef marine habitats can be crucial in the improvement of our understanding of ecosystems functioning, resilience, and how we can make future plans to protect what now has been mapped to its spatial extent in high resolution. These new data maps of the reefs within the Otuela MPA are aimed at broadening our understanding of these conserved biodiversity hotspots and the contribution of the abiotic factors, that is the seafloor and geomorphological characteristics to the ocean system's functioning. These variable seafloor structural characteristics are suitable for compatible spatial and temporal use patterns by specific species for their certain life stages. Multiple sounders is a great addition to the integrated approach of analysis and managing the marine space. At any scale, integrated analysis of the seafloor habitats is anticipated to improve the understanding of ecosystems functioning from a multidisciplinary perspective and management of marine space. therefore solving the lack of data-derived measurements of the biodiversity on the ocean floor. And with that, I would like to say, thank you.
Thank you, Tammy, for the very interesting talk. It was very cool seeing the bottom of the ocean. Any questions for Tammy? Philo, go ahead.
Hi, Tulwana, it's Matsubane here. Oh, sorry, give me a second. Yes, so I'm not sure if I'm understanding this very well. It was quite a quick one. But I wanted to ask, is Utrigela, the region, is it different? How different is it to either side, to the seafloor on Durban's side and on Richards Bay's side? Yeah, because from what I heard, I think there's a greater diversity of possible habitats, given that the geomorphology of the seafloor is very different. Like there's a lot of different patches. I'm not sure if I'm making sense.
Well, you are kind of. The fact that, as you can see that, as I'd shown with the examples, the visual differences in the appearances, that indicates that beneath the seafloor, there's also those differences. And with those given differences, as they can be easily visible or can be visibly seen, we can then denote from that they could possibly be supporting different kind of lives or different life stages. So as I picked the central and the northern and the southern, that was to show that across the span of the area, there's variation in the morphology of the seafloor, which then most likely would then support these several biological niches that I'm suggesting.
All right, thank you. That's quite interesting. I'm not well versed in geology, but that was interesting. Thanks.
You're muted. Thank you, Tammy. Do you think that you will make like a popular article about what we are finding in the sea? I mean, the bottom of the floor, ocean floor, so we can make it slightly less complicated for us who are not quite versed in the geomorphology.
Well, my intentions are to try and match my findings with some of the biological findings. Then in that sense, I will then try and work with the other guys to break down maybe some of the geological terms that are hard to understand, and then we'll try and infuse it with the biological terms and make more sense maybe to the biologically linked people. As I know, geology is not the stream that many people usually go towards, so it sometimes get a bit confusing if you want to try and break it down. And we in SAIAB, we have people like you who can always assist in breaking down things when we scientists fail to break them in a way that is better understandable.
Thank you very much, guys. Yeah, I think that brings us to the end of our session. I think the host will take over. Yeah, thank you, guys.
Thank you.
Hi, everyone. Thank you for sticking around for the afternoon. So I'm just going to announce our prize winners. And this year, the prize is a little bit different. The winners are going to get a customized coffee tumbler, which looks like this. This is mine. Can't really see in my... It's, I have Snoopy on mine, but the prize winners will be given the opportunity to design their own coffee tumbler. So that's the prize. Okay, so I'm going to start with the runner-up for Best MSc. Just give me 1 second to confirm. The winner, well, the second place runner-up for Best MSc is Vuyo Mko. Clap, clap. There's no clapping because we're on Zoom. And then the Best MSc, please feel free to turn on your microphones and clap. It feels weird. And then the Best MSc was Tembelihle Dube. Yay, woohoo.
Nice, Tembelihle.
Yeah, congrats. Okay, and then we'll move on to the runner-up best, the runner-up PhD. Sorry, I'm just multitasking like crazy here. And that is Mpilonhle Nyawo. Mpilo, well done.
Hello.
Okay, and then best PhD. Best PhD presentation, Nobukle Panza, well done. Woo! Well done to the 4 of you. I'll be in touch with more information about how to design your coffee tumbler prize. I think I'm going to hand over now to Nikki just to close off and say goodbye.
So after a very long day, I'm not going to keep anybody for long. I would like to extend a huge vote of thanks to my team, so Rebecca, Carla, and Pakama, for organizing such a great symposium today. Thanks also to Lucky, Vanessa, and the SciFest team. And I would like to thank our guest speakers who I felt all gave really inspiring talks. Thank you to the session chairs and our partner institutions. And lastly, but certainly not least, well done and thank you to all our student presenters. You have highlighted the amazingly diverse research work undertaken at SAIAB in freshwater, estuarine, and marine ecosystems. So thank you very much, everyone. Go and have a good weekend and a good Christmas break. Thank you.
Thanks everyone for organizing and presenting. It was great.
Thanks, Seascape team. That was awesome.
Thank you, Seascape team. Thanks, everyone.
Thanks, everyone. To all the amazing students, and thank you next to the Seascape team and Rebecca and Carla and Karma. Well done, guys.